Just a few words for those of you who are not that familiar with Biodiversa. What is it? What are we? We're a European partnership committed to the biodiversity strategy for 2030 and the vision that by 2050, hopefully, biodiversity and its benefit to people will be protected, valued, and restored, certainly all over Europe, but hopefully more broadly all over the world. We have broadly two groups of actors: ministries in charge of research and research funding organizations, and policy actors, ministries in charge of the environment and environmental protection agencies. Of course, like any large organization, we have a strategic research and innovation agenda. I'm not going into it. You can read all about it. It's 100 and something pages long. It can be found on our website. Basically, there are 5 themes, 3 topical ones: biodiversity protection and restoration, transformative change, and EU global action. And then there's the cross-cutting themes: better knowledge on biodiversity and its dynamics, which is where the monitoring comes in, and cross-cutting theme B, better knowledge for nature-based solutions in a global change context. Now, how have we implemented all of this? Basically, there are 4 what we call flagships. They're like large programs which combine several different activities on a theme. So we have one flagship on protecting and restoring biodiversity, one on improving transnational monitoring, promoting nature-based solutions, and supporting transformative change. And all those activities are divided in different work packages which focus on a certain aspect of the partnership. So work package 1 is support research and innovation. Then work package 2, for which we're here now, is support transnational monitoring schemes. We promote nature-based solutions and business valuation in work package 3. Very important for Biodiversa, but basically to improve biodiversity in general, is to have a good interface with policy and practice. That's work package 4. Work package 5 is international impact. Now, if you look at our budget, we have quite a sizable budget overall. From the EC, we receive 165 million euros over 7 years. That means that the partners who have to contribute 70% will contribute at least 385 million, which gets up to 550, but we know we'll get more. So we always say we're a partnership of around 800 million euros in 7 years, which is a lot of money to go to biodiversity. Just very quickly about one of our main activities. So, can you go back? No. How do I go back here? Now it works. As you can see in the budget, there's about a 1/3 goes to biodiversity monitoring and almost 2/3 goes to the work package 1, the calls, the research calls, which are our main activity. 6 calls over 7 years, 40 million euros each. So that's 240 million euros in research going to biodiversity. And we aim for at least 30 projects per call, all on a yearly schedule. The first one was on protection. The second one was on monitoring. So we have 33 projects which are looking at biodiversity monitoring from different aspects. Nature-based solutions was last year. Well, granted, transformative change is currently running. We expect to have the result by fall 2025. And then also in September 2025, we'll start a new call on the restoration of ecosystem functioning. And the last call we call future ecosystems for now, but it's yet to be defined. So I won't tell you about work package 2 because I'll transfer to Petteri for that. But just to give all the people working in Biodiversa their place, we have really large other key working areas. So nature-based solutions and biodiversity evaluation. There's a lot of activity around that. It's a very important activity within Biodiversa. Science policy interface, where we look at dialogue events and foresight, cooperation in knowledge hubs, and support by writing briefs and co-building the EU Science Service for Biodiversity together with the BioAgora project, about which we'll hear more as well. Internationalization is an important aspect. In fact, for work package 2 specifically, we're trying to involve more partners so we can cover Europe more broadly, but also look outside a bit to add partners. Communication is essential because otherwise nobody knows what you're doing. Stakeholder engagement is key, both for input into the partnership, but also to have them help with creating impact from the results from the partnership. And then you can find a lot of information on web platforms, which we're still improving yearly. That's all the activities except for work package 2, for which I'll give the word to Petteri right now.
Okay, thank you, Ron, for this general introduction, and hello, good morning, everybody on my behalf as well. So I'm Petteri Vihervaara from Finnish Environment Institute, and on behalf of Ministry of Environment, leading this biodiversity monitoring activities, work package 2. And this is our stepwise approach. So we have three installments during this 7-year period, and because this is really new thing for Biodiversa. It has been research funding so far, but currently it's also about biodiversity monitoring, as we heard. We have been really starting to discuss with our partners, Ministries of Environment and EPAs, that what we are trying to achieve and what kind of cooperation we want to do. The main starting period started with the setting the scheme to understand our common goals. Then we went to identify solutions and piloting different things for monitoring technologies and expanding our cooperation. And now we are starting to the end of the 7-year period and looking forward to the future, that how we can make this more institutional long-term activity for the biodiversity monitoring community. We have been working close cooperation with EuropaBON project in the beginning and the European Commission as well. And there is currently plans to establish and pilot European Biodiversity Observation Coordination Center, which is operating on the EU scale. But then what we are doing, we are working on a national, subnational scale, trying to support countries to establish biodiversity monitoring coordination centers on national scale, and then support long-term biodiversity monitoring with the harmonized methods and with the support of the novel technologies. This is the task and thematic structure of the work package 2. So we have priorities and common indicators, which are pretty much steering our work, what our partners and the countries are doing, where they are putting their effort at the moment. Then we have technical tasks about harmonizing of protocols and methods, database data interoperability, and then novel technologies and citizen science campaigns, how these could be also supporting long-term biodiversity monitoring. And today it's of course pretty much about these novel technologies because it's a science fair, and many of the projects funded from the biodiversity monitoring call are also dealing with the novel technologies. So from the work package 2, we really would like to work these projects to have a mutual cooperation and learn from you and get your expertise also to feed in for the work what we are doing in this work package 2. Then we go for the use of the data. So we want to demonstrate that the public data that is collected is significant for the society and the private sector and different players, research of course. And through all these, we have pilots running for different thematic topics. And the end goal is really to set up community national coordination centers and harmonized methods for long-term biodiversity monitoring. So all these different tasks are then feeding in for the final task, what is there as an end goal of this 7 years cooperation. There are a lot of things going on. I don't go through all these, but you can visit our web pages and go to the biodiversity monitoring, scroll down. There are all the deliverables and reports which has been done so far. There are reports about the data interoperability and protocols, priorities, indicators, and so on. So there are different toolbox. We have been working with surveys with our communities. Also, a lot of workshops and webinars have been organized during these years already, and more will follow. And some desktop studies has been done also about the understand and review some of the key issues like citizen science, usability, and the data infrastructures. And then we have pilots. And I stop with this pilot just to demonstrate that we have pretty much hands-on cooperation across 80 countries at the moment with the 6 pilots which are funded through this European Commission part of the co-funding. One was about the governance structure, which aims to support how these national coordination centers should be built up so that they are interoperable with the European Biodiversity Observation Coordination Center, hopefully emerging in the near future. Then we have a thematic pilots with different countries contributing and exchanging experience on invasive alien species, soil biodiversity, then automated biodiversity monitoring stations for birds, bats, and nocturnal insects. We have one focusing for rocky reef fish and marine monitoring, and then habitat mapping monitoring based on satellite and remote sensing techniques for grassland and wetlands. And many of these are in close cooperation with other networks and initiatives dealing with these topics. And currently we have the third wave of the pilots in the pipeline to be started next year. So there are 6 candidate pilot topics shortlisted from over 20 proposals that we got last year. So these are in the process, and 2 or 3 of them will be then funded for the next years. And these are pretty practical. We would like to learn what are the bottlenecks and the challenges if we want to make these schemes long-lasting and upscale them for the whole Europe. So there's a lot of like a assessment done based on these experiences from the different countries working with these topics. And that's all from my side. So thanks for coming here, and happy to discuss with your projects and over the science fair during these 2 days.
Good morning, everybody. I'm sorry a bit for the delay, but public transport always is a bit tricky in Barcelona at the morning. First of all, I want to thank you, all of you who come from different parts of Europe and who shows us that especially biodiversity doesn't understand about frontiers. And especially in a place where biodiversity is extremely endangered and it's a shared problem between different countries, we need to create these platforms in order to be aware and be capable to make decisions in order to fight this problem and to find some real solutions. We are really happy today to have this really interesting workshops and activities during these 2 days. We hope you are enjoying very much Barcelona, and you're enjoying very much this space, which is an ancient hospital. So we healed people, now we are healing nature. And we need to go further in this objective of making shared information in order to create clear decisions. That's why the government of Catalonia is fully committed with this objective, and will push on having the best database possible for our land, for our country, and for our Europe, which is the main objective of this journey and of these activities of the Biodiversa Plus. We would also like to let you know that we need this information to go further, to create some things which are, at our point of view, very useful to take decisions. That's why today I wanted to present you a bit an idea we had about 2 years ago, I think, about 2 years ago, and which has become today a real project that makes biodiversity so useful, not only for knowing, but also for being more efficient on managing and preserving biodiversity. What we had is a huge amount of information about species and habitats, protected areas, legal regulations, etc., which weren't connected. And we created a database, a common database, which is the CNATCAT, which is, I think they explained you before this database, or if not, they will explain it to you later. And this database contains all the information about biodiversity in Catalonia. But what we needed is to be more efficient on processing this information in order to have real solutions in the implementation of this information into real projects. You know that most of the time, when people, citizens, and enterprises ask us for making an activity to create a project, we need to analyze this information, and we need to find all this information in the different database, in the different laws, in the different sources, which take us some time and which can also create us some errors into collecting this data. So we decided to change a bit the way we, as public servants, interact with the citizen, and how can we go further on the knowledge of biodiversity and the interpretation of biodiversity, and giving time to this instead of giving time to things that are not useful for this purpose. What we did is basically we created an artificial intelligence tool, which takes the shape of the project they want to present us. In this case, we created with a race. I think it's a race, a bike race, or I don't know, bike race or a race, which is in a place. I think you maybe have known about it, Delta del Ebre, which is a natural park, and which has some endangered species. And what we created is this IA tool that crosses the different layers about biodiversity we have, the different laws we have, and also a database of old reports we made about this area, and makes us a draft which already shows us which are the main strengths of this race in this case. This gives us some time, instead of making this manually, to go to the field and to look directly what's happening and collect more data to put it into the machine. This lets us to be, as public servants, more near to the field, to be with more information from a basis, and without leaving any information apart because of the possibility of having a human error. This doesn't create the report directly. This just creates a draft. So the public servant always will be the one who decides and who has the last word. But we created a tool that gives us a lot of time to dedicate our efforts in analyzing the reality, the biodiversity in field, instead of crossing layers in the computer. That's why we wanted to implement some tools that demonstrate not only that biodiversity knowledge is important for having a cross knowledge and analyzing different patterns, but also that can be a very strong engine to give some solutions that are able to make biodiversity be easier to be considered in the different actions and projects we apply in the day-to-day of our country. So I hope you enjoy all these projects that we are going to see today, all the ideas that are going to arise. We, as government, fully believe you are power to our knowledge and to our capacity of evolving to a society who's able to preserve biodiversity as a part of its core welfare and well-being. And we want you to be happy today and to bring us lots of ideas. We will sure have the chance to receive it, and we will take it very seriously. Thank you very much, and have a good day.
Yeah, so happy to be here. My name is Blasi. I'm a researcher at CREAF and working at the EU project BioAgora, that I'm going to talk to you about it. So the title of my presentation is Biodiversity Monitoring Knowledge Network, It's abbreviated by CAN. So I guess we are not going to be the first ones to think about Barbie also when it comes to this abbreviation, but that it helps us to remember the name maybe. So together with Luis Brutons, also present today, and Sandra Luque, we are the co-leaders of this monitoring knowledge expert network. So we can first ask us, how do we organize all the data and information on biodiversity so that it's easily accessible for policymakers, given that nowadays the really big body of information is really difficult to keep track of everything, right? And how do we bring all the experts on projects together to discuss and produce impact from the research? So these are actually the main goals that comes for the BioAgora project and then the Science Service for Biodiversity. And the link is because BioAgora has the mandate to really produce the Science Service for Biodiversity. So the origin, yeah, I'm sorry for all the floating things that is gonna maybe prevent the slides to read, but the origin comes from the EU Biodiversity Strategy for 2030, which has the mandate that it needed to be produced a science service for biodiversity. So a really like the one service of science policy mechanism. And so then the mandate for it is the BioAgora project that started in 2022 and runs until 2027. And it's a Horizon Europe program with 22 partners in Europe. The aim is to connect the research results on biodiversity to the needs of policymaking. So we really work close together to the European Commission. So the main outcome, as I said, is to have really a fair and functional science service for biodiversity, the science policy interface at the EU level. We have many successful, like ongoing institutions and ideas on science service, science policy interface. Also, one example, Biodiversa Plus, but the idea is to bring them all together. So the mandate is to really work with EU-funded projects and to minimize the duplication of work. And we can say, we're going to talk about which ways are to involve them, those projects. So one way is, for example, to mobilize all these networks of knowledge and experts. And this is the knowledge expert networks that we were talking about. So the idea of BioAgora is that it organizes by several functions. And it's many functions, it's around 10 functions, but the idea is that they are engulfed in 3 main functions. So one is to really answer requests and building up evidence-based. The other one is to create the supporting active thematic networks and then transforming processes within and between science and policy. So the idea is that all these functions, they get sub-functions and they are all like interlinked together. So the BioAgora project aims at testing all these functions. So what works, what doesn't work to create this future science service for biodiversity. So I'm going to talk about one of the functions that they were mentioned, the answer request. And that's going to be linking us, how do we involve projects, for example. So the European Commission, for example, has a question, wants to know something about it. So for example, ecological habitat characterization of a certain habitat type. There is no information about to inform their policies. So then they submit a question to what it is the Knowledge Center for Biodiversity. So the Knowledge Center for Biodiversity also was starting in 2020 because of having the goal of implementing the biodiversity strategy. But then, it's hosted by the GRC. So the science service will be what it is. It's meant to be the scientific pillar for the Knowledge Center for Biodiversity. So then they have this question. Then they think if they can answer themselves via the GRC or not. Then they send it to the science service, which now it's being managed by BioAgora. So then we have, again, what we talk about this answering request support function, which helps by other functions, for example, the network. So we activate the networks given a certain topic, and then we want to answer that question. So then the idea is that the networks get activated, and then we reach to the knowledge community, which is, for example, through key relevant EU projects and initiatives. And then we create the expert group to synthesize and harvest the knowledge. If it's a knowledge that can be answered in a matter of weeks or a few months, so like 6 months tops. If it's more, then it goes to the CLIPS, which is already a mechanism that works. And then it goes back, and then it provides the feedback to policy to implement into policies. So then, for example, the networks that I was talking about, the current networks is marine freshwater, nature-based solutions, pollination, landscapes, transformative change monitoring, which we are the ones presenting today, and then more to come soon that we have. And the idea is that they are self-filling networks with a running science service for biodiversity. So the idea with the monitoring is that we activate the existing network of these institutions and projects in the field of biodiversity monitoring. So we bring them all together. And it's important to remember that, I mean, when we talk about biodiversity in Europe, it has to go hand in hand with EuropaBON and Biodiversa Plus. So we use the stakeholder network of EuropaBON, and a way is to test it during the ticket requests. And then we create, for example, links. We take on the work that was taken in EuropaBON on the monitoring network database. So to collect all the metadata of monitoring projects in Europe. And so far we have 28 projects mapped that some of you, we have already contacted you, that you are present today. Otherwise, we will talk to you more. And then we have policy relevant links in the EU, mainly with the EPES monitoring assessment, the Nexus assessment, and the scenarios task force. Also, we have links with the Committee of Earth Observation Satellites in the Biodiversity Expert Group. So just to the colleagues from EuropaBON also show us this, the slide of again, we all know what was EuropaBON, what was the main product of it to create the terms of reference for the EBOC that we're all waiting for. So we can also continue on that. But I wanted to just talk briefly about this webpage that contains the monitoring projects. So one of these, for example, analysis that we could obtain from that is that we evaluated all these 16 monitoring programs, for example, and how could it be the link to make them to produce EBVs. And in this case, they were analyzed 36 EBVs regarding species populations and community composition. And then we assess what are the bottlenecks on the workflows of the data flows. So bottleneck we understand as a factor that just prevents us the generation of this EBV at the desired temporal frequency and spatial resolution. So here it's just to understand a bit that we have the data collection, this is the raw data, then it gets integrated into a national or subnational node, and then that it goes into EU level. And that, for example, yeah, of course, some data can go directly to the EU level, and then we can produce these EBVs. And then, so for example, this is another way to represent it, that we can think about it, some stages that we go up, that we will end up with a final product there as the EBV indicators, that it was from data collection. So these are all the bottlenecks that prevent us from passing to the next stage, or that reducing the information that we're gathering to get these EBVs. So for example, if we lack of monitoring guidelines, we don't have a standard monitoring scheme, we are really getting some data that cannot be really integrated later on. Or if we get lack of fundings, so that it can affect on several of those stages, as well as, for example, an monitoring initiative that it's at integration at the EU level. So just to show you an example how we evaluate it, we have these different stages of data collection, models, and data integration. And we evaluated if there was a no bottleneck, partial bottleneck, or a full bottleneck for each initiative. And that's a way to, with a flower visualization, running, and then we can evaluate in a really quick look. I mean, there is a lot of abbreviations, and I will happy to talk to you about it later as well. But we can see, for example, here that we have not so many problems regarding data collection groups, and then more problems maybe when it comes to data integration. So for example, the partial match of some monitoring programs with the definitions of the EBVs, and in some cases, there is no matching at all with the definition of the EBVs for that to produce that EBV. So the main bottlenecks, as I said, they were related to data integration. So that refers, for example, open code, software, or funding. The list of data collection, as I mentioned, and then also a matter of tackle those bottlenecks collectively. So for example, funding doesn't mean that we only prevent from having that monitoring program, but it also affects on how many volunteers we can coordinate, how capacity build is done, and how the IT infrastructure. But this framework can also be applied. It doesn't need to be in the EU level, but also it can be in the national level. And yeah, that's it. I'm done now. And but just to say that the coming activities with the CAN monitoring is to continue working on these links of EBVs and the nature restoration law, habitats, and bird directives to help on the reporting, and then to expand the network that I just showed with the monitoring database, keep supporting EPES, And we're going to plan a project event early 2026 with links with the EBOC with support of the Commission, and we will continue answering the request. So thank you very much. We are here, Luis and I, and then also Sandra is online. Thank you.
Hello, I'm Michael Cunliffe, here from MARCO-BOLO project. I'll talk about it after the coffee break. Biodiversity data flows, we're also doing that in the MARCO-BOLO project. How much crosstalk is there between this project and other projects that are happening right now?
Yeah, so thank you for the question. So this is, of course, like a work that was done with EuropaBON, and we are extending it. So we are really revising it. And what I shown here was like some of the results on terrestrial monitoring programs. And of course, from this evaluation, the one that was really lacking was the marine system. And we're really aware that MARCO-BOLO, for example, this is key, for example, on helping on with this initiative. So the idea is like to really now expand to reach out the current projects of who's doing what, and then how we can together like build it up much more completely. So, and of course, some of these some of these workflows were also set on like in situ data. So of course, like it's not maybe taking into account other forms of data gathering. So we will have to maybe develop other frameworks, or how do we check from other projects, how do they do it? But the idea of the CAN monitoring, for example, is to bring together MARCO-BOLO from the marine ecosystem with another project that deals with these problems at the terrestrial ecosystem, and then to bring them together to discuss, to see what's the problems, what are the solutions, and then to really offer this opportunity of discussion, of coming up with solutions rather than they're sitting all in their own on their own projects. So that's the idea. Yeah, thanks.
I think you already answered my questions before I could ask it, but okay, let's go again. I think this is great. It's nice to get this overview because indeed there's so many initiatives on the national scale, regional scale. I really hope that maybe starting today, after indicating the problems, which are well listed, great summary, and we all can probably nod, but to also look forward to how do we then solve those issues? Because I think that's what we really need, the pragmatic solutions. How do we overcome that we don't have data standards quickly without talking for another 5 years?
Yeah, definitely. I guess you answer also yourself. And
no, but is it part of this project?
A bit the sky's the limit, but in a more funding limits. But it's more like what the projects are interested in, what can we have the capacity to organize, and of course the idea is to solve the issues once we have them identified. No, but of course this is complex. We see how much we can solve, and we will continue working on that. And but yeah, the idea is to help. Finding the solution. It's always easy maybe not to identify the problems and then not do anything about it. But I guess that's what I put it here as well, to think about maybe with also new technologies. Can we tackle some of these bottlenecks? But that's what I said as well, that we also don't have to be blind of tackling one of the problems, because sometimes it's like by tackling one of them is an umbrella also, or like a chain continue of bottlenecks. So yeah.
Okay, there we go. Good morning, everybody. My name is Päivi Sirkiä. I'm from Finnish Environment Institute, Sirkiä, and I work as a senior specialist and group leader. So great to be here. Thanks for the organizers for this possibility to talk about our project. I can see that the slides are a bit different what it should be, but let's see. So our project, Biodiversity Life, it's about prioritized actions for promoting biodiversity targets in Finland. So our goal is to halt biodiversity loss. And this is a huge project nationally, and we act in different ways. So for example, we improve biodiversity conservation network and restore habitats. Those are the expensive big parts of the project. I'm gonna skip those today. And we also develop data management and data availability and mobilize biodiversity funding, plus then also improve cooperation networks and enhance know-how. And that's the part I'm concentrating on today. So yeah, as I mentioned, it's a huge project and very many project partners, which is great. Several ministries, governmental organizations and non-governmental organizations together. And yeah, the work package that I'm leading, it's about coordination and development of biodiversity and ecosystem monitoring. So our goals are to increase openness, findability, and interoperability of nature-related data. And we want to strengthen the cooperation between organizations and experts that are producing, delivering, and using this important information. So in practice, we develop coordination and interoperation of biodiversity monitoring schemes, and we also develop existing monitoring networks, some of them, and then we mobilize new monitoring methods. And I'm going to talk a couple of words about all these three in the following. Yeah, so when we start to think about the coordination and interoperation of monitoring schemes, so it's maybe good to remind ourselves about the situation. So we have several organizations that are running monitoring schemes, producing data, and there are a lot of voluntary work also involved. And there's pretty much no overall coordination over this. And when it comes to the datasets, many of the datasets are not easily findable or accessible still. And of course, it's always so that there are still many aspects of biodiversity that are not being monitored at all. So yes, a lot to do. And the first thing that we have done, and we have started, so we have put up a cooperation network of habitat and species monitoring scheme experts from the most central organizations, first of all, in these biodiversity monitoring schemes. And actually, this theme group is part of a bigger network in which altogether 17 organizations from diverse sectors have united to enhance the quality and accessibility of nature-related data. And this bigger network we call National Coordination Group for Nature Information, and which is coordinated by Finnish Environment Institute, and our chairperson is Petteri Vihervaara, whom many of you know also from Biodiversa Plus. So this bigger network has 4 different theme groups: data utilization, monitoring, innovative methods, and data interoperability. And this monitoring theme group is integrated in this Biodiversity project. And so what we are then doing actually is that we have started to put up a roadmap for biodiversity monitoring nationally. So first thing was to review the current monitoring schemes, which we have actually done in a couple of former projects. So we have reviewed altogether 90 systematic species monitoring, and then we have put together information on all these biodiversity monitoring schemes on a web portal called Finnish Nature Information Hub, which is actually also a metadata portal and also exists in English. And the second thing, what we are now doing is that we are reviewing how well do we meet the monitoring obligations, such as reporting for directives and agreements. And for that, we are identifying data gaps and gaps in data management, and then at the same time, try to figure it out, what are the possibilities to fill these gaps? For example, could there be some synergies between monitoring schemes, and what are the possibilities for new methods to fill these gaps? And then we aim to make some prioritized proposals to improve these monitoring schemes and data management. So yeah, well, it's important that we do this together because, well, together we achieve more. And I actually think that together is the only way that we can agree on this kind of roadmap for biodiversity monitoring nationally. And then I will hurry up to the next part, which is development of existing biodiversity monitoring networks. So in this part, we concentrate first of all on implementing complete species lists for observing species among various taxa. This is actually pretty new in Finland. We are not used to using checklists, but I hope this will spread from now on. And we are also improving metadata and sampling design for certain monitoring schemes. And in this, we focus on breeding birds in protected areas and moth monitoring and raptors. And at the same time, we are also improving the data accessibility of these mentioned monitoring schemes. The third part is developing of new monitoring methods, field monitoring of habitats, and remote sensing-based monitoring and molecular monitoring methods. So in short, well, this is a clear data gap we have. So we should have more systematically collected field monitoring data on habitats because the national forest inventory we have, it only covers like a bunch of the most common habitat types. It's not enough. So what we are doing now is that we are planning a detailed data collection protocol for essential biodiversity variables for each habitat type. And also at the same time, we improve data management practices. So the plan is that we will continue this method development and field testing a couple of more years. And then about the same for data management. It's actually quite a big thing for an ecologist. It's a surprisingly big thing. And then after 2 years, we should have the ready method and we could start monitoring. But there is still this question mark because we don't have funding for the monitoring yet, but the need is anywhere super clear. And then just quickly about the remote sensing-based monitoring methods. In this, the idea is that multi-sensor approaches will be developed to detect spatial patterns of ecosystems and habitat types. And in this, the focus is on peatland habitats. There have been some piloting done already on undrained peatlands mapped in the selected pilot sites. And well, in this, the focus is on collaboration between biodiversity experts and remote sensing experts. And last, well, stop, says Iris already, but I will continue by 1 minute. So yeah, we develop also molecular monitoring methods. And the first task in this was to review the technology readiness level and possibilities of the methods, which was really nicely covered by the recent article by Laamanen et al. Of course, this was also something that was started in former projects. You cannot put such a review together in 1 year. And shortly, the main 3 points in this work has been that well, there is still a long way for many of the methods. And the bottlenecks seem to be the lack of large-scale collaborative efforts and lack of standards. But we should still put up large-scale pilots because we cannot just wait for the perfect methods. And well, we should also prioritize monitoring and development to serve the knowledge needs for policy landscape. And yeah, the next step is to choose 3 to 5 specific methods and pilot them during the remainder of the project. And well, in very short summary, in Biodiversity Life, we develop biodiversity monitoring coordination, develop existing monitoring networks, and implement new monitoring methods. And together, we simply achieve more. It's important because we are super scattered, at least in Finland, but this is the way to go. So I want to thank you for your attention, and special thanks to all these people who I have been working with in this project and many former projects. Thank you.
Thanks. Thanks, Päivi, for presentation. My question is, do you know if there are similar live projects going on in other countries like this?
Well, good question. And I don't know, but if somebody knows, I would be super interested to hear. And any kinds of similar projects.
Good morning. I believe that your work is fantastic, and I would like to congratulate you on this. Just a brief question. Is it possible, you think, to exchange knowledge and expertise, and you could bilaterally help other countries to broaden their horizons on this, just like you do? Thank you.
Thank you. Good point. Well, we would like to learn from each other. Yeah, I think that's the way we should do, like share experiences and learn from each other.
Good morning again. Hi, everyone. So before starting the section 2, there is a lost mobile phone. Seems it's coming from someone from JBIF. I don't know. But if someone has lost his mobile phone, please ask me. So let's go with section 2. I'm Danny. During this second session, I'll be introducing the representatives of 6 cutting-edge European projects that are pushing the boundaries of novel technologies. We will also have time for questions after the presentations. And a quick reminder about the marketplace. The marketplace is open and ongoing during this session, so feel free to explore and connect between talks. Thanks. The first talk is from the ARISE project and will be presented by Elaine van Ommen Kloeke, the program manager from ARISE, Naturalis. So Elaine, so the floor is yours. Thank you.
Hi, everyone, and thanks for joining this session. If you need to remember anything from my presentation, it's one thing. We do species recognition at scale, and we'd like to do that for any species. It's a national program, so we're only solving this for the Netherlands right now. But of course, like most of you will know, technology doesn't care about boundaries, neither does biodiversity. So I really feel that we can actually translate this kind of technology and everything behind it to other countries as well. Right. Basically, we have 2 big streams of work. We do identification based on DNA, and we do it based on sensors. And you see many of the sensors that we use also in the marketplace today. All in all, so we have a future where we can complement the work of the many ecologists in the field by automating some of these kind of observations. Of course, that also means that you need references. So it starts with building a reference database. You can't do eDNA monitoring if you don't have the specimens, if you don't have the barcodes to identify them with. Neither for AI. You need to know which species is which. So that's one of the core activities actually of my program, and we've been running now for 4.5 years. So we've been quite busy. It also means that we need proper workflows, end-to-end workflows that can quickly capture metadata, specimens in the field, work them up to a particular degree so they can have a barcode, generate the barcodes, do the blast results, get the data back to the person that collected it in the first place. And not just 100, right? Not just a research scale kind of hundreds. We're talking about 100,000 per year. Automation is therefore key. And also in the lab, we've actually acquired a lot of big machines, fancy with little lights on top, that do the job for us. And what you see here, for instance, that every specimen that comes in, either that's collected by an expert or a Malay strap, gets an image. And a 96 well plate is being done now in 15 minutes. So you grab a cup of coffee and you have over 90 pictures done to microscopic scale. It also means that you really need to have a grip on your data. The biggest question that we're still struggling with is what species do actually occur in the Netherlands? Do we have a specimen of all of them? And that means connecting all kinds of data sources and knowing, do we have enough? Because we want to have 3 specimens of each species. That's also making sure that you communicate to the outside world, we don't need this specimen anymore. Please focus on the ones that we don't have. So what you see here is actually a user-friendly version of what we call our gap analysis or target list. I'm going to see if I can hide this. Oh, not really. There we go. That's nice, right? You can actually see my slides. So not just a big Excel file, not something in GitHub, but something that's interactive. So on this list, you can type your taxonomic name and it will have the taxonomic tree behind it. And you can search, is it wanted? Is it unwanted? Do we already have data in bold, for instance, or data ourselves? And it really helps us to drive our experts in the right direction. So no more butterflies, please. We have those, but please go for the midges because there's many and we still need barcodes. It also means standardization. And again, apps that are user-friendly. And I'm trying to convince my experts who are usually 70-year-old experts that don't like computers necessarily, that want to do it their own way, but we still manage to convince these grumbling old lovely men, usually, that actually it helps because now we can capture their data on the mobile, on the tablet, immediately in a standardized fashion, making sure that the data run through the computer easily and they also get their results back easily. And that's been a phenomenal change. So I'm going to do this again, people, sorry. Biodiversity also means that you need to adapt and be flexible in your protocols in the lab. So we've now have, of course, the insect protocols, which were out there already, but we also adjusted for plants who are very hard, fungi that sometimes need maybe up to 10 different markers to distinguish a species, but also how to work with collection, so old DNA material. And we are now also merging our efforts with the eDNA world so that we have all of these protocols in the same lab and we can actually learn from each other, but also lower the costs. Which of the parts, which of the parts of these protocols can we actually do ourselves, right? Not using the big company protocols because they're expensive, but lowering the cost so that any colleges can actually make use of our infrastructure. And we save time. The big machines are not just fancy, but they actually save us a lot of time as well. So extraction protocols make sure that we do plates per minute. And we even don't exchange the plastics anymore, which also has a nice sustainability part of it. So it's been a real nice effort. And like I mentioned, we're talking about 100,000 specimens per year. So this is our achievement for last year, and we're ramping up those numbers even further by specializing. And it also means those apps. So here's a full circle of our whole going from specimen in the field all the way back to giving the results back to experts. We have interfaces where they can log in and they see their own results. And I think from a citizen science perspective, this is a big hurdle to take because everyone wants to work with us, but they don't want to wait for their results for 3 years. And that's been kind of where we were. Like, we don't know what to do. It takes time to get the results back. But now they can actually view like a track and trace of your package, your post package. Where's my snail? Did it get sequenced yet? Is the result as I expected? And return feedback. The second component of Arise is more to the sensors that you see here today and the AI involved. One of my teammates who's here, she's actually the expert. So if you have any questions on these sensors, feel free to ask them during the market. We've tested more than this, actually. We've tested over 20 different sensors in the field, and many of them are here. We brought the Diopsis camera, so over 300 devices have been in the field, over 500 deployments, millions of images, terabytes of data. For instance, for the wildlife cameras alone, we have areas where 65 cameras would be in a given area to answer ecological questions. And so over 1 million images, all of them need to be annotated. Not all the AI is there yet, but we do our best to improve that at the same time. For the Diopsis camera outside, we have been in the field for 4 years. So over 31 million images are already collected and processed. That also means that you need variation in your AI. So instead of having a single AI work on the sensor, we actually build a platform with a repository where all of these AI can be collected and can be used actively. So that means we can not only analyze this, but all types of sensors. And we really invite the computer science domain to actually add their AI. So it's not lost in a publication or a repository, but you can select it here in an interactive interface. You get your data in, you select your AI, it runs, and it can process a lot, because that is one of our biggest core elements, enabling large-scale automatic species recognition from any camera or audio sensor. And that means that, for instance, last year, it took 7.5 hours for 26,000 media items, currently 15 hours for many more. This is the short list, and here you can already see that we also collaborate with different initiatives. So Mambo is mentioned here, and I've actually forgot to mention Biodiversa, because also some of the sensors are, of course, through Biodiversa collaborations. Last but not least, the interesting part. How do you manage all of that data? So I promised a bit of this. Because we have such a variation in data, you need a proper data management system. You need data models. You need a system that creates lakes, that connects the data, and then also pushes it out to all the platforms that you need to have it on. Not just talking about repositories like GBIF, but also your own interfaces. And so we are creating now domains of types of data. We have a DNA domain and has a data model. We have a media domain, but there will also be a GIS domain, a species domain to tackle the taxonomies behind it. So to validate and harmonize the data. One of the struggles that it will tackle in the harmonization is, for instance, here you see an expert in the field will collect something and the name is given of the collector. But if they turn it into a sheet, that same type of record is actually called recorded by. Our data management system allows that harmonization of that data. So even if different terms are being used, we can now connect the dots. And that's necessary because an ecologist and biodiversity needs more than a sensor, needs more than one method. It is the combination of all of those methods that will give you the biggest view on your biodiversity and start answering those ecological questions and compare the methods as well to benchmark. They also want to have a secure environment. So this is one of my last slides. We have a login system. So only you can access your data with a login and you decide when you push the data or give it to someone else to be shared with. And that should somewhere around October all come together in our nice looking platform called Arise. So I welcome you to test it in October. And it's also part of our biggest strategy. Definitely as an organization, we're building on top of each other. It's not just this one. I will also be speaking about BMD. So we're basically stacking all of these different programs to not stop after 5 years, but to continue build on top of the innovation to come and harness that in that organization. Thank you.
Thanks, Elaine, for this inspiring project. So we have time for 5 minutes for questions. So the floor is yours, please. It's open for.
Yeah, I was wondering if the tools you're developing are open source, either code on GitHub, or is it propriety of a Arise project?
It's a mixture of things. So as we are also government funded, of course, we are as open as possible. In some cases, we're still building. So putting it open right now isn't necessarily the best approach, but it will be open once we're ready for this. In some cases, we also use commercial products like data storage. Of course, everyone knows they probably need Amazon at some point, even though we don't want it. But for the data management system, for instance, we use Databricks. It's a management system that really helps you to orchestrate your data. On top of it is Airflow, which is again then open. So it's a mixture of all of these kind of data technologies.
Yeah, hi, thanks. I would like to ask two questions directly, if it's fine. So the first one is you have many different imaging systems for the field, and you talked about the 96 well-played pictures in the lab. But for larger insects, do you also have then different cameras in the lab, or how does it work?
Yeah, it's not operational yet, and I only got 10 minutes, so I needed to, I was already quite fast. Yeah, we're also what we call a grid stacker. It's a very unsexy name, but it's the same for pinned specimens. So these are the dry specimens where we basically have built a rig system. It's quite simple, where the camera basically moves around these items and also makes the stack images per item. We're still figuring out how to automate this because you still need to pin all the specimens and then you have the little cards underneath. So we're figuring this out. And we want to put AI on top of it once we have now that data together as well. But this is for the future. Yeah.
Yeah, thank you very much. If it's fine, I would directly ask the second question because of the sustainability of the plastics. This is not only for the reuse for imaging, but also the reuse of plastics for DNA extraction.
Yes.
So you bleach them in...
I think it's actually done with fluorescent radiation, maybe. So this is the machine is called a tip washer, and so basically the whole thing goes into a module. I'm just a manager, so you know I don't know all the details. And so it rinses them in some way so they are usable again and again and again.
Yeah, that's perfect. I try to convince people also to do this then in the other lab. Thank you.
Yeah, just another one. So what are you going to do with the data after the project ends? Because I guess it's really a lot of data. So it's like terabytes and terabytes of data. So when the project ends, there's no money for the storage. So like, unless it continues, but how are you going to tackle this?
So my message and vision that I would really like to emphasize here and to all of you and Biodiversa, we should try to work into visions where the projects support the vision and not the opposite. So I think our biggest hurdle is also that these projects are ending and the data and knowledge is lost. In my case, I'm lucky that our organization believes in this. So I say ARISE will never die. There will be additional programs on top and it will be integrated into the organization to maintain these systems. So it will not disappear at all.
Thank you.
Thanks, Elaine. So let's go for the next talk about the Mambo project. So we welcome Kim Bjerge from the University of Aarhus, representing this project. The floor is yours. Thank you.
Thank you. Thank you. Yes, my name is Kim Bjerge, and I'm working together with Toke T. Høje, and we have been working together for 7 years now. And the project I'm going to talk about now is the modern approaches to monitoring on biodiversity founded by you. And the project has started in 2022 and it's running one this year and next year. And there's a number of countries, Denmark, UK, the Netherlands, Germany, France, Malta, and Bulgaria that's involved. We have 8 work packages where Toke is responsible for the whole project. And it's really about finding new technologies and working with them to see how we can actually monitor habitats and species with different methods. The objectives, there are some mentioned here in green, which I've chosen to speak about. The others are in progress. So we'll work on different ways to use images and sound to monitor species and habitats. We are using high spatial resolution images, and we are also looking for some of the habitat condition metrics using LiDAR and drone data. We are working on standardizations for habitat metrics and using deep learning and remote sensing to monitoring different aspects of biodiversity. Some of the knowledge gaps we are looking at is the gaps of insects, taxonomic, how actually monitor insects in real life, bats and amphibians. The tools we are going to use is computer vision, acoustic, and LiDAR methods. And myself is specialized in computer vision and deep learning methods. I'm an engineer, where a lot of the people here in the project, of course, is biologics and ecologics. We are working on doing also digital infrastructures and having a lot of activities and stakeholder engagement. I would like to also emphasize this paper we have made towards a toolkit for global insect biodiversity monitoring, where it's focusing especially on insects. And some of the objectives, the first one using image and sounds. As you saw before, with the Arise, we have already made a model for bird songs recognitions. And we also have made generated a species list for plant communities. And I'm especially been involved in these automatic moth monitoring of nocturnal insects with this trap here. This is one of the first versions. And we have been working on a pipeline that actually running on the trap, and it has been running now for 4 months without any problems on 12 traps in Denmark. And it's doing first detect the insects in the image. Then we have a broad taxon classifier that classifies what order, suborder level it belongs to. And if it's not an insect, it's something blurry. And then we have a moth species classifier. And then we also do tracking, because the camera actually records a small video sequence of the flying insects. And so we get a lot of data out of it, where we can see about the taxon, the species, the confidence level, the date, the time, the duration it was observed, and a lot of information. We're also working on satellite images using radar to get climate data from the satellite and combine it with the RGB images to make maps on the bioclimate time series, where we are using the UNIS habitat hierarchies to classify the different areas. And that's made a map for Europe about that. There are also an activity on habitat metrics using drones and LiDAR points to measure details. And Daniel Gislinger has just published a paper on, and I really recommend if you are into this topic here, where he's comparing a lot of different methods and what you actually can get out of the drone data, where you have a lot of details about the habitats. Some of the questions for species detection and classification, which I'm working on bats and on insects especially, we will dig a bit into that. So one of them is to detect insects in the field, and we have 2 here. It's specialized bird cameras that's also good for insects because they have a close focus range. So they are mounted about some sedum plants that's flowering during the year with different flowers. And we have actually been working with this in other projects also. And we have 6 sites in Europe where we have situated these cameras. And here currently we are working on a classifier that has a hierarchical classification of family, order family and species genus. And it also has some uncertainty saying whether it's not sure what it is. And here's an example of what we can get out of it. We have actually 48 cameras mounted for 2 years in a field and grassland and different areas. And here's an example of what you can get out of it. So we also made an algorithm that can detect the amount of flowers, because that, of course, is very important when you are monitoring a small square of a typical 50 times 50 centimeters. And then you can get nice plots of the activities here. I've selected pollinators, spiders, grasshoppers, flies, arthropods, and ants. And we are also now doing ecological studies to can we see something about the abundance, species, community, how it differs during the seasons. And the same for the nocturnal insects. We have here data for 3 years. Here's an example of plot for one trap, and you can also observe that the abundance of the different order of differs a lot. And if you dig into the data for the Lepidopterus, the micro and macro moths, you will actually see that some of them are really very few times during the year they occur. But you can search on it on this one, and you can actually see a nice video on YouTube if you like to see what the algorithm does. We also been in Mambo projects, been working on how can we optimize the manual way to do plant analysis, and that is using either drones or the street views. So you have a camera filming when you're driving in a car on the motorway, and AI Lab is presenting an example of that system. And the whole idea is to detect invasive plant species. Here in Biodiversa, we have used some of these tools in 2 sub-projects, invasive alien species and automated biodiversity monitoring stations. I'll speak a little about them. So we have deployed them in a lot of different locations around Europe. And the latest we have done is that for the Amy traps made in the UK, I've taken all the hardware out and replaced it with some we have built our own, also on a Raspberry Pi, but our own software. We've added a modem. And the nice thing is that I can sit on my telephone and support all the traps around Europe. And I can also change the software. And what is running on the trap is this algorithm I showed you before. So data is uploaded, not all the images, they are also uploaded, but also the nice report of what is in the CSV file with the tracks of the species and all that. So you can use that for further analysis. I think that was all I had.
Thank you, Kim, for this amazing presentation. There is time for questions, so floor is open. And even if there is some question from the online audience, please ask, feel free.
Thanks for your presentation. I was wondering, given that there are a number of devices that are being developed and used in different regions, in different countries, there are also a number of different data sets that have been gathered to train the AI to detect and classify all of these insect targets, these nocturnal insects. How useful, in your opinion, would it be to pool all of these data on a European or maybe even a global scale to do one model rules them all? Or are you more advocating for local models, for local solutions? Do you think we're very far from that, or do you think it's even useful to go in that direction?
It's a good question. It's one of the hard things. Well, deep learning is really easy. It's just to train a model. The hard thing is to gathering the data and get them cleaned and ensure that they are right and they have a high difference. So my experience is that if we look at here, you can see that they used one data set for training the localizer, one data set for training the moth species classifier, and another data set for training a more overall classifier. And my opinion so far is it's a really good idea to divide your algorithm in several steps. I know that algorithms today can do everything in one, but it's very hard to create a data set on a big image that contain all the species that exist in the world. So I suggest something like that, what I've done here. And if we look at the moth species data set, it's really trained on data from GBIF. So it's just downloaded all the species that exist in UK and Denmark. And we have, so GBIF is a good place, but my experience is that data recorded with a specific camera differs from these data. So it's really good if there are also some data from the camera. The data that's trained for the broad taxon classifier is based on data from the trap. And right now for this problem here, I'm using data on the trap, and I'm working on an algorithm now that had 80 different genius species collected from the traps. And I'm working together with researchers in Germany and France to help me sorting them out. But it would really be nice if we have a system where data is collected and somebody sorts them, and we could reuse that for training new models. That's really the most important thing, I think, that is finding a way to how to do it. And they're different from museum species and species collected here with the camera traps. You can see them, they have different backgrounds. They are blurry, they are more difficult. And some of them you cannot classify to the species level, only to the order level or whatever. So the next talk is about the MARCO-BOLO project and will be presented by Michael Cunliffe from Marine Biological Association. Welcome. The floor is yours.
Hello, thank you everybody, and thanks for the opportunity to talk about MARCO-BOLO. Also to be really depressed because I'm from the UK that is not part of Biodiversa Plus, and coming here today, it's all this amazing stuff making me feel terrible. Anyway, enough of my problems. So I've been asked to give a short summary of MARCO-BOLO, which is Marine Coastal Biodiversity Long-Term Observation. It's a Horizon Europe project. We're over halfway through. We end next November 2026. And there's a few of us here I'll mention towards the end. So I think this infographic really sums up what MARCO-BOLO is about. But there's probably no major surprises for most people in the room. Marine ecosystems, certainly coastal marine ecosystems, are in trouble. Lots of pressures, lots of stresses. There is a demand to improve monitoring. I feel like right now we have all the bits and pieces that we need to put together, but the key is bringing these things together. So the different monitoring approaches, the different data streams that people have talked about already, and then how do we put those into packages that are useful and understandable for a range of different users? And where do we want to be? We want to be in a place where we've got healthy, beautiful, and resource useful marine ecosystems like these ones in these pretty pictures. So we were asked today, MARCO-BOLO is a large project. It's got lots of elements to it, different types of technology, a big emphasis on data. We've lots of talks about data this morning. I'm just going to focus on some of the environmental eDNA work that we're doing in MARCO-BOLO and try and give you really some specific examples of work that's happening right now. So hopefully most of us know what environmental eDNA is. We've been really trying to validate environmental eDNA as a monitoring tool, but particularly focused in marine and coastal biodiversity observation, which just as a sort of observation of Biodiversa Plus is maybe not so much. A range of different things really listed here. We've been doing some meta-analysis of pre-existing studies. So, I mean, I've been working on eDNA now for nearly 20 years. People talk about emerging technologies. I sort of feel like it's been around for quite some time. It's part of my PhD. So I think lots has been done, but it's sort of sporadic and really needs to be brought together, and we're doing some of that. I can't believe we're still doing this in some sort of ways, but we're comparing traditional techniques with eDNA-based observation. Just to say, I really don't like the word traditional. It often gives this sense that it's old and then outdated. So even though I say traditional, I don't really mean it. Developing operating standards, and I get particularly that I think is quite useful is taking the data sets that were generated from eDNA and actually making them into useful things, particularly EBVs and indicators. So, I mean, this is hopefully it's a conundrum that we're still in, and it's a bit depressing that in 2025 we're still sort of having this problem, is this performance analysis between eDNA-based techniques and traditional, in inverted commas, and still having questions about, are we measuring biodiversity equally? Are we getting more? And something we don't really talk about so much, but I sort of feel like it's a conversation we should have, is about, is it easier and is it cheaper? I mean, I'm a optimist and I always look at the best in things, but the amount of heavy and heated discussions I've had with traditional biodiversity observers about molecular tools who get exceptionally angry, and I really mean the word angry, about eDNA. And so there is a lot of pressure around it. One of the, I'm trying to pick out the real highlights. One of the highlights, I think, in MARCO-BOLO is looking at eDNA with long-term data sets. And eDNA long-term data sets are actually quite few and far between. And I think that's maybe a unique aspect of what we're doing in MARCO-BOLO. So some of the work that I'm involved with, so this is a long-term ecological time series station. We call it Station L4. It's off the coast of Plymouth. It's been studied for more than a century. My institution's been studying it since the turn of the previous century. But what we have is a really unique 20-year eDNA time series, and they're not so common, these multi-year, multi-decade eDNA time series. We've been using that to look at, to compare, I guess, eDNA with traditional. Just to mention some fungi, we were having to chat about fungi in the coffee break. This data set actually comes from another project looking at fungal diversity within coastal marine waters, which is effectively impossible without eDNA-based approaches. One of the major groups of organisms we're interested in MARCO-BOLO are plankton, coastal marine plankton. And one of the things we've been doing is comparing eDNA-based approaches to look at plankton, particularly phytoplankton, with, again, traditional inverted commas. And what am I talking about? A taxonomic expert who's been doing it for the last 30 years, looking at down a microscope and identifying specific types of plankton. And basically we come along and say, well, we can do your job on a Friday afternoon. And perhaps not surprisingly, they get upset about it. Just a snapshot of some of the data sets we've been generating. This is just one example of many different types of phytoplankton. This is Pseudonychia. It's a diatom. It's particularly important because it produces a neurotoxin called domoic acid. So people interested in this from a harmful algal perspective. On the far left, we've got the microscopy data from the 20-year time series. And on the next to it, we have the metabarcoding from the 20-year time series. And we can see this is, of course, I picked a really nice example of where eDNA and microscopy actually works quite nicely. Some work that we're literally just about to start. We're just about to send the DNA samples off for sequencing. We're really interested in plankton, but also working with other partners within MARCO-BOLO, particularly the Arctic University in Norway. We're also going to start looking at fish. And again, one of the nice things about having data from this time series is, as well as the long-term eDNA data set, we also have long-term traditional fish data. So these are fish caught in nets, trawl nets, but also fish larvae and fish eggs. And so we think this will be a unique comparative data set for fish eDNA, metabarcoding, but also fish, I guess, traditional again. 3 minutes, okay, thank you. We're interested in long-term observation. We're also interested in large-scale spatial observation, and probably one of the best marine large-scale spatial surveys was the Tara Ocean expedition. I'm not going to mention this too much because Pedro Junger's in the audience. Pedro's going to talk about this, I guess, later in discussion or speak to him. But as well as long-term time series, we've also been looking at spatial scale work. Generating these data sets, they're really nice, effectively lists of diversity, but one of the really important things now is to move into taking eDNA-based data sets and generating indicators. So a major activity of MARCO-BOLO has been looking at EBVs. And again, Pedro's here. Pedro's been working on this on the MARCO-BOLO project. So maybe grab Pedro if you want to talk about EBVs, and he might mention this in his discussion session later. Particularly around eDNA and plankton, we've been exploring to what extent we can use metabarcoding for indicators, and some of the indicators for plankton are used in things like MSFD, so measuring good ecological status. So here's just some examples here, these different pelagic indicators. But if you actually look at what these indicators are, again, 2 examples, the ratio between pelagic diatoms and benthic diatoms indicates storminess, or the ratio between holoplankton and myriaplankton. I'm pretty confident we can actually do these with our metabarcoding based data sets. And what's quite nice is within the project, we have Claire Ossel, who's at my institute, who actually developed this technique. So it's the plankton life form extraction tool, the PLET tool, which people use for microscopy. So we're seeing if we can use it for molecular as well for metabarcoding. And I'm pretty confident in a lot of it. So there's a few of us here. So I just want to mention Pedro and Emily. Pedro's going to do a discussion session at 3, where some of us are involved with a topical panel on eDNA. You might have seen our poster already, but if not, please take a look, because we'd quite like to get your perspectives on eDNA as well. Just to finally, this is the MARCO-BOLO project. It's led by Nick Page and colleagues at EMBRC in Paris. Lots of names here. These are most of the people involved in the eDNA-based work, led by Chris Bowler and Daniel. The partners are there, and I've got 1 minute left, so I'm slightly ahead of time. I'll stop there.
Thanks, Michael, for this interesting talk. So we have some time for questions, so the floor is open.
Oh, sorry, I'll repeat. eDNA can only happen with reference data. So I'm wondering what you're using and if you're producing any references yourself.
Yeah, so for the plankton metabarcoding, we've principally been using this PR2, so the PR2 reference data set. And that's been interesting and useful. For the fish, we have there is a curated, it's ridiculously called the British Fish Reference License, that somehow fish know what the boundaries are. But that's a real bona fide, in the same way that you talked about for references. But we hit the same block that I think your problem is dealing with. So one of the groups I'm interested in is algal parasites. So these are eukaryotes that infect algae. And for most of them, the reference material isn't easily available. So each individual parasite, we have to go into the literature, find the reference barcodes and do that. But my lab has been involved with the Darwin Tree of Life project, which is sort of similar to ARISE, generating reference barcodes for all the eukaryotes in the UK, sort of slightly ambitious, I think. I don't know how many eukaryotes you have in the Netherlands, but
not that many, 47,000.
Yeah, so it's very ambitious, but we've also been accessing the reference barcodes from the Darwin Tree of Life project.
And the data's open?
They're accessible. They're not widely available yet, but we were a genome acquisition lab.
I'll send you an email.
Yeah.
And maybe for the experts that are seeing their jobs disappear, it might also be nice discussion also in this group here. What I felt is I have some of them that act as ambassadors. So I make them present what the benefit is to them as a taxonomic expert, because they basically feel great. They don't have to look at all the plankton anymore, but they have the cherry picking and discovering of new species. So these kind of arguments actually do help.
Yeah.
But you need to put them in that position.
Yeah, I mean, I come from the microbial world, so bacteria and microbial eukaryotes. So we have to use molecular tools. The idea of looking down a microscope and figuring out what bacteria there is absurd. And one of the things I was very naive with the sort of plankton community, the sort of light microscope plankton community. And yeah, and I totally get it. We are literally talking about someone's profession in theory. And so I just think certainly everything I've seen so far is a requirement for compromise and complementarity. And I can imagine a place relatively soon where the large scale bulk work will be done with eDNA, but we'll still have to have that sort of reference approach. Yeah.
Thank you very much, Michael. So let's go for the next. OBSGESSION. The project OBSGESSION will be presented by Maria Santos from the University of Zurich. Welcome, Maria. The floor is yours.
Thank you. Good morning, everyone. It's my pleasure to be here, coming from the remote sensing laboratories at the University of Zurich to present our project, OBSGESSION, Observation of Ecosystem Changes for Action. So the project meant to respond to the call from the Horizon program on interdisciplinary assessment of changes affecting terrestrial and freshwater ecosystems, building on observation programs. It has 11 project partners listed here. It covers 7 countries. It has 4 years of duration, started last year, and we have 2.5 more years to go. And it has about 7.3 million in terms of funding. So what are the objectives of OBSGESSION? The objectives are to monitor and predict biodiversity change and its direct and indirect drivers in both terrestrial and freshwater ecosystems. And we aim to achieve these objectives through the integration of state-of-the-art multi-sensor Earth observation data, innovative in situ, including citizen science data, and next-generation ecological models that account for uncertainty. Where does this come from? Well, you probably are familiar with the proposal for a global biodiversity observing system that is needed. This is an example for Colombia, but there is a big set of scientists that say that regional biodiversity observation networks are necessary that integrate multiple of the data sets that we've been hearing about, including also Earth observation. Also, there's been also a push forward. What is it that we understand about monitoring, and what are the drivers of the changes that once we monitor lead to these changes, and can we act on those? There has been proposed a framework for modeling what we call detection of change, and then attributing that change of biodiversity. And then finally, this monitoring and this understanding over time of the drivers and pressures might help us also understand biodiversity processes, which can go from evolutionary legacies all the way to human modifications. So within this context, OBSGESSION has 5 objectives. The first objective is to look at current science and policy needs. You're all familiar with all of those, but we wanted to assess how can these new technologies and new data platforms respond to particular relevant EU policies and directives related to biodiversity. Second objective is to look at what is existing in terms of Earth observation, citizen science, and in situ data to assess biodiversity change and what are the drivers. Then we want to understand, so using innovative models that are emerging in ecological sciences, different types of modeling of ecological processes and biodiversity change. Objective 4 is to look at then closing the gap. We started with the gap with what is needed from a policy perspective. So how can science actually provide solutions that are uncertainty aware to cover these gaps that are identified in the policy? And then finally, of course, dissemination and share the findings in as much as possible within as much actors and interest groups that we have. So OBSGESSION aims to fill in specific needs. The first need is harmonized biodiversity monitoring. This should not come as a surprise to all of you. And I also put there the definition of an essential biodiversity variable, just to refresh people of what are we talking about when we're thinking about monitoring this. Within OBSGESSION, we did a review of the European Union policies and where do the EBVs actually are called in and can contribute to certain types of actions. What we see is that EBVs can be applicable broadly to 37 of such actions. And these include all the 6 EBV classes and 21 of the candidate indicators of EBVs. But policy needs, they can be divided into 4 groups. So is it to review, to report, to plan, or to implement? So then we also reviewed which kind of processes can EBVs, and here's the list of EBVs, be useful for the policy to implementation process. And you see here the first results on why do we need actually this harmonized biodiversity monitoring. But then moving on, we also want to respond to the use of improved Earth observation data. So how are we planning to do this? We're planning to create a lot of data cubes, both from in situ and Earth observation data. And we're also trying to assess a seamless transition to upcoming new sensors or downscaled sensors coming from airborne platforms. And I will talk more about this in the afternoon on the applications of these new airborne sensors. What are the EBVs that we're focusing on? We focus both on terrestrial and freshwater systems at the EBV classes of community composition, ecosystem structure, and ecosystem function. Each one of the groups and the partners has specific foci, but then overall we are producing data cubes that will enable to develop some of these biodiversity products and ultimately lead to the EBVs. So how does this work? We have on the left, on your right-hand side, what are the data cubes? We want to integrate them together in time series products that are zoomable to different local needs, and they are easily extractable. And then we want to provide EBV-enabling data cubes to the users later on that have specific needs. We're also focusing on expanding to the new upcoming missions that we see in the Earth observation and space agencies portfolio. Here's an example of imaging spectroscopy in the optical domain. We have already a couple satellites with this technology, and we will see more in the future. And what we're experimenting with is what kind of information on biodiversity will these new platforms actually provide to us? And what is the uncertainty associated with these observations? I've heard a couple of people mentioning the word uncertainty, but in OBSGESSION, we're really focusing on how do we actually propagate the uncertainty from the Earth observation, from the biodiversity in situ observations into the models of biodiversity that we then want to use to inform policy solutions. And this is the third objective of OBSGESSION is to actually deliver knowledge on the drivers of ecosystem change. How is this knowledge going to be delivered? We're using a set of AI and causal inference and causal discovery, new types of models that enable us to look at temporal changes, use counterfactuals, and then assess what might be driving these changes. We also have a set of biodiversity pilots, 6 of them specifically, that cover these freshwater and terrestrial environments that I was mentioning. We have grasslands, we have forests, we have different freshwater ecosystems and estuarine ecosystems. So the expected results, so we are year 1.5, are to first to provide a framework for detection attribution modeling. So this will enable us to understand the direct and indirect drivers of biodiversity change. We will provide harmonized biodiversity monitoring approaches and products, pipelines, biodiversity and data, biodiversity enabling data products and data cubes. We will look into improved predictions of ecological transitions and tipping points using these new modeling techniques that enable us to experiment with different conditions. Scalable cloud platforms where all the data will be openly available, improved uncertainty assessment of biodiversity, not only at the collection state, but also how does it propagate to the models and the predictions that we make. And then, of course, close up the gap that we identified first. What are the science policy recommendations for prioritization of certain actions for biodiversity protection within the European realm? So with that, thank you very much for your attention.
Thank you, Maria. So we have time for questions, please.
Yeah, thanks a lot. I'm Felix from Rabobank, a Dutch bank that finances farmers all around the world. You mentioned that you provide suggestions for restorations. Do you also have a focus on avoidance of, well, not in a restoration context, but in a, well, what is not typically a protected area, but outside the protected areas, which parts would be best to avoid and kind of harmonize that corporates can use that to see, well, where to focus on what is best to avoid in the current practices?
So thank you for your question. So we have 2 models that we are starting to use and we'll be using the next 2 years of the project. The first one is using the EONIS habitats that we already seen and looking at ICLU, so a land use modeling, to see what kind of options will be there to avoid certain land use types and certain land use modifications into the future European landscapes. And then together with WCMC, we're using a restoration model as well to predict what would be the future areas for restoration and also to assess whether there are areas that could be avoided for modifications.
Okay, let's move forward. Next. So for the next talk, we welcome again Elaine van Ommen. She will talk about biodiversity meets data from Naturalis. Thank you.
So I'm giving this presentation on behalf of my colleague Niels Raas, who couldn't make it to this meeting because he has a GBIF meeting in Ireland, I think, or where is it? No, sorry, Waterford. So forgive me if I don't know everything about biodiversity meets data. It also just started, so we're really in the starting phase, but it's super exciting. Mostly because this is a large EU-granted proposal, and it's actually not inventing anything. It's basically, there's also the slides are here, so there's a few additional ones. It's a huge horizon call. And it actually has many partners that have already developed a lot of things. And you'll see and recognize many of the logos here because they're involved in your projects and your initiatives. So yes, we are working with LifeWatch and Catalogue of Life and Pansoft and the Royal Gardens and GBIF. And it's basically seeking out that combination of what we have already today and make it into something pragmatic that we can use. So Maria, I had lots of questions for you, but I didn't want to raise my hand all the time. But let's talk about these data cubes. It also means that we're trying to connect multiple programs. I remember the question on the rise, so who's going to keep this alive? Well, this is exactly the point. If we're going to stack programs, then we're probably going to make it, because now we can combine the outcomes of particular programs and build on top of it. So what you see is that DISCO is involved, ARISE is involved, the Biodiversity Genomics Europe is involved, which will probably get a next phase as well. MARCO-BOLO is involved. And how can we combine that all together? So what's the big idea then? Now, of course, we're all working against this state of the biodiversity. Can we plan and manage and expand protected areas? In a nutshell, how can we equip the field managers so they can manage their areas well? That starts with better monitoring. So here is also where ARISE enters and some of the eDNA programs enter. Can we make it simple so that these managers can direct the questions from the bird directive, the habitat directive, the water framework, the marine strategic framework, make it quick, easy, and simple for them to monitor in the field if there's no available data yet. Across all the Natura 2000 areas, or at least as many as we can muster, so that we have a clue about the state of our biodiversity in those areas and can take action. Yeah, and of course, all because we want to be net positive at some point. So, first and foremost, how do we get those high throughput methods in the hands of our field managers? And can we then also mobilize, of course, historical data? One of the presentations already said, well, there is data, but it may not be as accessible. So how can we make sure that the data that is already being out there is accessible to anyone and everyone for references, but also because the measurements are already there? And get them into the big biodiversity repositories, please, please, please. Creating a data catalog. What data is already there? Can we combine the data and move that towards a platform? So also a pipeline to harmonize that spatio-temporal data and the taxonomy behind it. Nobody mentioned taxonomy yet today. That's interesting because it's one of those big gaps, right? Yeah, I see a few nods. We can make this interactive. Who has an issue with taxonomy across countries or in a country? Ah, see, there you go. There you go. And I'm sure that many of you have opted this also in your project proposals to try and address this, right? Yeah, same here. So let's find each other because I think we're trying to do the same thing and we should be able to tackle this 200-year-old problem, right? Last and not least, citizen science. How do we actually engage the community? How do we engage those experts that have traditional or manual labor that now need to go into a future where they can use tools and don't feel obliterated out of their expertise, right? We need to take them along and show that it's complementary, show to citizens that they can also be part of this world using particular tools. It's not an easy job. And last but not least, an access point. Yeah, basically creating the platform that combines that all and makes it accessible. And training, capacity building. Apologies, it's a bit of a dry presentation because it's just started and it's basically just listing what BMD will do. But we're starting up fast. And so hopefully in the next iteration of such a Biodiversa meeting, we actually have some cool results to show you. Just quickly running through all of this. So this is the bigger picture, right? The data services on top with the platform and then all the given environments. So the data access and mobilization capture, the data harmonization layer, the data storage and visualization computing, the virtual research environment. This is where the data cubes come in. And of course, then the platform itself where you can access. Oh, yeah. Oh, love this. So if you have questions, and I am not sure why these images didn't come through, but if you see all the sensors out here in the market, if you have questions about this, please reach out, because I think we can harness a lot of that information through the Mambo network, Arise, and Biodiversity Genomics Europe. It's copying a presentation that wasn't checked. All the data will go to GBIF. So regardless, most of this data will generate an observation, and obviously the observations will go to GBIF. Of course, we will check if there's no sensitive species and not post those, but if we can, we can. For those that do not know GBIF, hands up if you don't know GBIF. Nobody dares. Okay, good. Billions of records, and it's growing by time, I think. And GBIF can also help you mobilize that legacy data. Well, then the gaps and the cubes. So the data harmonization is something done together with multiple initiatives again, building these building blocks so that we can answer particular data question, combining GIS data, combining habitat data, combining species observations into these cubes that are then easily usable for other measuring and modeling. It also means that we want to visualize. Also something that sounds very easy, but isn't necessarily very easy. We need to engage with what does the end user want to see? What's useful for a field manager versus for a policy manager versus a researcher, and how can we combine that? And make it fair, obviously. And connect through APIs or other data measures through the VREs. These are the virtual research environments that are out there or being created as we speak. And that's it. Most of it, I think. There's a few more, but I think we're there. Yes.
Hello, thanks for presenting again. I really like your style of presentation. But I also have a more general question. It seems like we all want to achieve the same thing. We're all headed the same way. Harmonize, collect, unify, all that. And I'm just wondering, after listening to another of these presentations, is it really useful that we have so many initiatives going on? Is there anyone, any institution out there that can really, maybe in a more authoritative way, tell us to join efforts? I mean, I work more on the methodological development of biodiversity monitoring methods, and this is often something I advocate for. Diversity is useful. It's useful to have a number of approaches and solutions, but in the end, we also need transparency and to avoid redundancy. So what is your take on that?
Yeah, nice question. Thank you. Almost philosophical. I feel that if you see development, you always have this storming phase of lots of innovation, things happening at the same time. If I translate this to your daily life, yes, I have Netflix and Disney Plus and Amazon Prime, sorry. They each still have their own little thing that is unique, but you also see some of these things then disappearing over time because it's kind of merged into a particular system. I think there should always be a level of diversity, so you have a choice. But this whole storming phase is super useful because then you can develop a lot of technologies, see what works, see what doesn't work. But I agree with you, in the background, you do need to see this buildup of solid repositories, solid initiatives, and less competition between each other. Yesterday, we had the workshop, and one of the big things that will make it a game changer is if we don't fight each other, but find each other through these big initiatives and infrastructures. Because many of these projects, indeed, what happens after? So we are focusing on things like Catalog of Life, GBIF, the BOLT systems. My own institute is really big time investing in this whole data management infrastructures, its core infrastructure with AI modules so that it stays alive and we can facilitate, hopefully also on a larger scale. But even we can't do that alone. It would be much better if each country had such initiative, either distributed or not, that's okay, but at least build up the core that we need.
Thank you for the presentation. And it was actually very interesting. Like the question, I also had the same, and your answer was interesting. So I hope that you're right, like it works that way, because I was also thinking why so many initiatives doing the same stuff, but I see your point. And I have just a funny note. I don't know when News was preparing this presentation, but now the occurrences in GBIF are more than 3 billion, not 1.
Yeah, exactly. So that's already an old image. Thanks for that. Yeah, it's super. Yeah, like I said, I'm a big advocate of GBIF. I know that some of you are in GBIF nodes in the country, but I think these are one of the big initiatives we should all support because that's the open data that everyone benefits from. So yeah, thanks.
Hi, I have a question kind of bouncing off of that. If all your biodiversity data then goes to GBIF, which is amazing, what will that single access point then bring on top, or what is that giving access to? Is it a new platform that then will give, I don't know, what does it give access to?
I'd like to see at layers. And what I've learned also yesterday sitting with more policy, and I'm more in the research realm, is ultimately, policy wants to have output on the EBVs, right? The essential variables, do the CRCD compliance at companies, and then we're at the research level generating the data. In between, that needs to come together. So I see GBIF as a repository of generating the data, whichever means possible, and of course, whatever quality possible, so there needs to be a check. But at least you have a repository where you can then go beyond the observations and maybe connect that to abiotic data or a GIS data or the remote sensing data. And then you start to make predictions about the ecosystem, about what's the state now versus what it was last year or next year. So all of this, of course, needs context. And I think the biggest achievement is not just, do we know which species exactly lives where? That's great, but you will never get there. But can you compare situations and see that you're going towards a positive direction or a negative one?
So will it then also use other data that doesn't come from your project, but just all of the GBIF data?
So biodiversity meets data will it will also use GIS data. I don't know exactly which data, but I'm assuming multiple data sets that they then form in these cubes that Maria explained so well, that you can then use for particular digital modeling. Yeah, absolutely.
Thank you.
More questions? No? So thank you. So the last talk of this section will be about the project Seaghost, another fancy name for a project of new technologies, and it will be done by Raúl Ramos. Welcome, Raúl. The floor is yours.
So first of all, thanks a lot for the invitation and allowing me to present this project. So the Seaghost project, well, I thought it was a big project, but having seen yours, it includes up to 17 partners. The project is coordinated by the University of Barcelona. There are other 10 partners that receive funding. And also there are another 6 self-funded partners. Okay, you can see that the partners came across all Europe and also include 2 partners from North America. The project is just in the middle of the implementation, and the budget it's over 2 million euros. Sorry, and it received funds from several national funding entities as well. The aim of the project is gathering knowledge on the spatial distribution and the trophic ecology of storm petrels, which are poorly researched seabirds. One of the big aims is studying the migrations or the annual distribution of these birds, assessing how the populations mixed, so gathering information on the migratory connectivity of these populations, and also studying the at-sea behavior of these tiny birds. Second key objective would be establishing the conservation units of these birds that they breed in Europe, as some of them are not very well defined as species. And then also a third objective would be overlapping the several human activities with the distribution of these birds to assess the human impact on them. So the project currently includes 6 species of these storm petrels, currently recognized species. We will discuss about this later. And the outputs of this project would interest different actors that are interested in marine biodiversity and also marine spatial planning, okay? From the academia to policymakers, also the industry, NGOs, and the entire society. But what's the relevance, which are the relevance of these storm petrels? So currently, most of the seabird studies have been always based or biased toward the larger species. Here I just included a picture to show you an albatross, also a shearwater or a petrel, and then on your right side, a tiny storm petrel, just to show you how small are they. So my size lower than 15 centimeters, just to have an idea, like a swallow, something like this. So the project will provide this knowledge on the spatial distribution, which we believe could be the last frontier for studying these seabirds at sea. Here I just put also a device to show you the size. I think they are circulating a bit, some of them, just to show you how small are these devices that we are currently using. You could be able to check them. So these tiny devices that allows us to track the migrations of these birds, we call them geolocators or light level geolocators. The weight of these loggers are very small, lower than 0.5 gram. Okay, and we tested some years ago, 3 years ago, successfully with a population of these storm petrels in the southern Spain. And then in this project, we aim to distribute these loggers across all the areas where these birds breed. I will not get into much detail on how these loggers work. So they are not GPSs, so they are light recorders. And from this information, we can estimate the location, more or less the location with a large error, as you can see here. We can gather 2 locations per day. Okay, just analyzing the light intensity stored in the loggers. Also, a handicap is also that we need to recover all these loggers to get all the information. So the spatial scope of the project, so as I have said, it includes 6 different species. In the first plot, you can see the breeding colonies, known breeding colonies of the European storm petrel, which is a very clear species. Also, the second plot includes 2 different species, the Liger storm petrel and the white-faced storm petrel, breeding in the northern countries and in the Macaronesia respectively. They are very differentiated, these 2 species. But in the third plot, we can see 3 different species which are very closely related, and they are often mixed in a complex of species belonging to the band-ramp storm petrel complex, okay? The distribution of these last birds are from subtropical and tropical areas. I just came here with a short example on this last complex of species I have just mentioned to show you more or less what we would like to end the project with. So here just to represent, so there is a picture of one of these birds breeding, in this case in the Canary Islands. An example, also a picture how the loggers were deployed in the leg of these animals and when we recover some of this information. Okay, I will show you more or less later on the output. So here just also I came with a single example. So this is a single trip of one individual breeding in the Canary Islands. So we recovered just a few months ago. You can see the breeding colony marked with a star and how this animal migrates towards the North American coast to the Gulf of Mexico and also the coast of North America of the United States. So it's a bit rare because most of the migrations are often latitudinally, but in this case, this bird or this population, as you would see, migrate longitudinally towards the coast of North America. So this is just one example, okay? But the project, of course, would consider much more data. In this case, these are somehow preliminary data. The idea of the project is to include much more data. The sample sizes that you can see here are rather small, you could see, but still allow us to to see different patterns of migrations across these taxa, okay? What I wanted to say, yeah, in blue, you could see the northern populations that breed in the coast of Portugal and also in the Canary Islands. The two populations all migrate towards the coasts of North America. The birds from Azores, which is considered another species of storm petrel, most of them migrated towards the coast of Brazil. You can see it in yellow. And then the other populations that we included in this preliminary analysis, birds from Cabo Verde and also from Santa Helena, from the southern hemisphere, more or less they mixed in a common area in the south central Atlantic, okay? As I said, this is the sample size might be still small. This is another way to show more or less the same results, to show how these populations connect. These migratory populations connect. At the top of the circle, you can see the breeding origins of these birds from different populations, and at the bottom, the destination of these birds. Okay, this plot is interesting also because it's standardized or it's corrected by the population size. So basically, the larger the arrow is, the larger the population of these birds. And you can see basically a couple of things here. So that in the Gulf of Mexico occur different populations, including birds from the Canaries, from Portuguese, and also from Salvagens, which are some Portuguese islands close to the Canaries as well. And also another interesting point is that in this South Central Atlantic wintering area, so many populations aggregate, including birds from Santa Helena, all birds from Cabo Verde, and also a very few individuals from Azores. So in fact, from another species, or what is considered now another species. Okay, and finally, I also wanted to highlight that only birds from Azores, so it's the Monteiros storm petrel, all or most of them wintered in this coast of Brazil, in southern Brazil. Okay. Okay, and to finish, so this was just to explain you a bit about the first aim of the project, that we wanted to assess the spatial distribution of these birds. But the project would also like to include diverse data, not only spatial data, but we would also like to compare the size of the animals from different populations to see if differences arise between populations. Also analyze the diet of these populations, if they differ, playing here or analyzing the elementary composition of the diet. And finally, also record the voices of these birds and see if their calls are different from others, okay? Basically to see if they are able to recognize. Okay, and I have to stop right now. So thanks for your attention, and any question, let me know.
Thank you, Raúl, for the presentation. So we have time for questions. Floor is open.
Other species?
Sorry?
Can you scale your technology to other species?
Sure. So, so the devices that we provided, I don't know if they were stuck somewhere. Okay. So yeah, can be applied to many other species, including terrestrial ones. Okay. The ones that I show you has an extra layer of plastic just to protect them from the salt of the sea or from the water. But yeah, they can be applied, and they did so many studies using this technology. Yeah, sure. So the thing which is a kind of novel is the size of this or the weight of these loggers, because maybe 5 years ago wasn't possible to track these birds because of the size and the weight of these loggers that were only light enough until recently.
More questions? So I have a question. So what is more or less the recovery rate of these loggers? So
that's a very good question and a tricky one. Yeah, it's so basically depends a lot on the colony. So for instance, this first attempt that we did in 2021, the recovery rate was 90%. So we deployed 10 loggers and we recovered 9 of them. But other colleagues from the northern countries, Sometimes they deployed 15 or 20 loggers and they recovered none of them. So it depends a lot on the colony. Also depends how the colonies are managed. So for instance, if you handle the birds from the nests or you just capture from nets. So it's a very different rate what you finally get. So if you basically deploy the loggers on birds breeding in the nests, you ensure that these birds are breeding there and probably they will come back later the next year. If you do it by using nets, for instance, you are not 100% sure that you will find the same bird next year.
More questions? No? So thanks to everyone. So this section is ended. Thanks for the great presentations and sharp questions. It's time to refuel. Lunch is at the garden.
Okay, thank you everybody to be here in this the best time of the journey to listen people talking about sounds and monitoring. So thanks to be here. We will try to be as fluent as we can in this time to be, don't worry you. So this is the first topical session where we are going to try to discuss with three great specialists on the topic about ecoacoustics and the potentials and threats of this technology. As you know, passive acoustic monitoring is basically we go into the field, we put some recorders that we can set up on continuously or programming when we want to record. So we can go back on the field, we go to the lab, we download all the data, we use different techniques of artificial intelligence, we try to interpret the sounds and we identify which species or we have recorded. So this is basically what we try to do. Is something a technology and emerging in some of the ecosystems. But for example, people from the sea or people that is working in bats, they have a long history of that because they have other limitations to monitor some of the species. And of course, basically, we have to focus on species that they can produce noise. So that's one of the best limitations. I would like to introduce here as in the Catalan partner of the Biodiversity, some of you, we send you a survey to know which is the state of the art of the implementation of some of the novel technologies in the different partnerships. And we ask you for different technologies, and I will try to summarize some of the results from a roadmap that we have. It's already published in the web page of the Biodiversity Plus. And we have seen that ecoacoustics and use of passive acoustic monitoring is among one of the highest level of development among Biodiversa partners. So there is a lot of partners using them, basically on mammals mainly bats, but also other species and birds. But there is also incoming and there is a potentiality for using on other taxons groups like amphibian, reptiles, some kind of invertebrates, where the academia is already developing technologies to use that. One of the interesting things is that it's used in a very widespread type of ecosystem from sea, from terrestrial, even from freshwater environments. And the EBVs, the type of more monitoring EBVs that they have been using the partnerships are basically as for the species population to know species distribution and somehow abundance, but also to study phenology and movements in some case. So it was one of the methods that they tackled more different EBVs. So it shows that it's a methodology and a technique that has a lot of potential, but as an emerging one, we have identified a lot of problems already in the full implementation. Most of the reported problems that the Biodiversa Plan partners has arise were related to data storage, analysis, and synthesis of the huge amount of data. I think it's something very common in all these new technologies. The cost, mainly in the initial cost and also in the time processing. Some partners, they issue about technical difficulties, probably linked to the lack of capacity and training in some of these techniques. So I would like to stop, and with this summary, I would like to introduce the panelists of the roundtable, that they are the ones that really know about that. And I would like to introduce, first of all, in my left, Kevin Darras. He's a researcher on the French National Research Institute of Agricultural Forest and Environment, specializing in ecoacoustics and automated biodiversity monitoring. He leads the Worldwide Soundscapes Project, a global initiative compiling passive acoustic monitoring data across terrestrial and aquatic ecosystems. He has developed innovative tools like the open source platform EcoSound Web for analyzing ecological soundscapes. And he is working in integrating technology with ecological studies and advanced sustainable agriculture and biodiversity conservation. More to my left, I would like to introduce Benjamin Cretois. He's a researcher in NINA, Norway Institute for Natural Research, specializing in integration of artificial intelligence with ecological studies. His expertise encompasses AI-driven image and sound classifications, cloud services, statistics, and all these kind of more techie issues. He's now coordinating together with Caroline at Biodiversa project, TAPMON project, towards a national acoustic biodiversity monitoring program. It's one of the BioMod projects. And the central objective of TAPMON is to develop a transnational biodiversity monitoring with autonomous acoustic sensors across a large latitudinal range in Europe, from Spain, Catalonia to Norway. And further on the left, Adrià Baucells. He's an ecologist and researcher in the Museum de Ciències Naturals de Granollers, here very close to here. His work has been focused on bat conservation and acoustic monitoring and impacts on habitat disturbance and bat populations. He has done a lot of, he was explaining all the countries has he been working. It's amazing. Madagascar, Amazonia, Brazil, a lot of countries. But now he's coming back to home and developing all the research here in Catalonia. And he has been working very hard in the last year into integrating all this acoustic data to generate indicators of bats populations, and also working at European level on how all these initiatives can be integrated in all the data of acoustic data of bats can be integrated and at the European level. So just to open the floor to the panelists, I would like, and taking the ideas that we have identified on this survey, we would like to focus the roundtable in 3 topics based on recorders and deployment. The second one, we will talk about data analysts, and finally, how we communicate all this data to stakeholders and managers. And finally, we will give the floor to the audience to interact with the panelists. So going back to the first topic about the first step, we need to put recorders. There has been a huge advances on recorders. In the science fair that is already ongoing, you will see a lot of different models of recorders from bats, for birds, from very expensive, from very cheap, in common, Creative Commons, open source area. So there's an emerging technology on that, but this is also, it's an opportunity, but it can be also a problem because there is differences on the accuracy, on the quality of the recording. So all these things, and the technology is advancing very quick. So, and this can introduce inconsistency in data quality and comparability in long-term studies. And when we want to analyze multi-site analysis with different types of recordings, with different type of settings of programmation of the recorder. So this can be a trap for the robustness and repeatable of the data. So I would like to give the floor to the panelists to ask them if, given these technical and logistical hurdles, how can we move toward standardized protocols for device calibration, deployment, and data management that are both practical and scalable across diverse bioacoustics initiatives? So the floor is yours, and you can talk about that. Kevin, for example.
Maybe I can risk an answer. Thank you for the question, Gerard. I think there are many ways how passive acoustic monitoring for bioacoustics and for ecoacoustics can be done. There's a variety of devices that are out there, of deployment settings and strategies, and a variety of ways how the data are used. And if we want to achieve standardization, we need to do that at several levels. There are many aspects of the entire workflow that can be calibrated, that can be measured, that can be standardized. And so, for instance, one of the questions I get the most often is, especially when people start in the field of passive acoustic monitoring, they're asking like, well, how far does my recorder record sound? And then I'm like, no, that's not how you should ask the question. It's more about the source. How far does the sound of the source, so of what you target, how far does it go? And actually, it depends on a lot of things. It depends on how loud, of course, the source is, where it's placed, what frequency it's using, how high it is, whether it's on the ground or in the canopy for a forest, whether it's in the superficial aquatic layers or more in sound conducting channels. All of this exists, and it can be quite complex to actually answer that question. And this is something peculiar about passive acoustic monitoring because compared to other methods where we know from which area or from which volume we're sampling, with passive acoustics, it's actually quite tricky. So we need to measure this. We need to calibrate our devices. And this is one thing that I'm really advocating. We need to know how large is the space that is sampled. And we have techniques for this that I'm not going to go into technical details, but this is one first way to standardize the data. And it's required to actually make comparisons between sites. You can't just compare the sound you recorded in a grassland to the sound you recorded in a forest, because the detection spaces are just not going to be comparable. And of course, there are, so as I said, there are other aspects that on which you can work to standardize these data, to make use of these data and to collate them into syntheses and to ask large-scale research questions. There are a lot of initiatives that are ongoing to make sure that just the metadata, so the descriptive data for our projects are gathered and reported in a standard way, just so that we understand who is doing what, where, and when. We're still working on that. There are several initiatives. Benjamin is going to present some of his work. There are initiatives in the ocean, in the aquatic realm. And we've also been working with the Worldwide Soundscapes project. We have compiled data sets from diverse ecosystems to show that it's actually possible. It's not just, we don't want to just standardize for the fun of standardizing. We're doing this so that we can actually put all this data together to ask bigger questions with larger data sets. And so we've started doing that, and we could actually show that once we gather all the data from even terrestrial ecosystems, aquatic, marine, and freshwater ecosystems, subterranean ecosystems, we can put all of these data together, and with a pragmatic approach, we can analyze them. It's important to note that there are different communities also that are maybe more or less good or advanced about standardizing different aspects of this entire passive acoustic monitoring workflow. For instance, as terrestrial ecologists, we could really take a leaf out of the book of people who are working in the ocean, who regularly calibrate their recorders. We don't really do that much in the terrestrial realm, or who have advanced models to understand how far the sound is propagating. All of this is still nascent in the terrestrial ecosystems, and I think that this is something we should more go into. And yes, just to close my answer to this, I think this is actually a requisite to all those goals that we mentioned today, to bring all the data together and to work together. We need to have comparable data sets. And this is why standardization, even if it may sound tedious, is required.
Yeah, thanks. That the thing is critical, because the people we are working on passive acoustic monitoring, we say, okay, that's a good, because you will have long-term data where you can go back and reanalyze the data of long-term years ago. But if we don't have all this metadata on how it has been recorded, it's a critical issue. Thanks. And of course, yeah, we say we get a huge amount of data, but then how we process this, and as other methodologies with image in the RISE project, we are really focusing on developing AI models to classify these sounds, to link these sounds to a specific species or behavior of one species, or even we can individualize somewhere even the individual if they have some kind of clues. But one of the things that we have seen that there is a lot of initiative. Every team is developing their own artificial intelligence models with different technologies, and the ones that they have been using 2 years ago or 3 years ago, probably we have a much better ones, new models, new neural networks models to develop. Everyone is adapting or creating their own model for a specific taxa, for specific environmentals or questions. So this can hinder cross-study comparison and collaborative scaling, because at the end, it's very difficult to share the raw data. We need to share the outputs of the data in somehow, because so this is a key point. There is also, we have seen it that there is an interest of building shared platforms of acoustic analysis and pipelines to streamline and democratize across different users, from citizen science to governments to even researchers to tackle with this diversity. And one of the things that we also is critical is that, okay, when you run some of these models, they give you outputs, a lot of outputs, but how trusty are these outputs? How we make this kind of validation? How we check the trustness of this data? So it's very important, because at the end, if with this technique we want to be impactful, we need to be trust on what we are saying. So in this context, I would like to ask to some of you that, do we face the same analysis challenges across different group taxas or not? And what steps we can take to build trust in bioacoustics data while moving towards a more harmonized, interoperable system and data analysis and sharing? So it's something that it's a critical point also, I think.
Yeah, I think I can answer that. Just to link back with what Kevin said, I think especially for AI models, like the most important is to have data that's machine readable in a way, or data that have metadata that are kind of common in a way. So like, I think it's really important that when people start a bioacoustic project to think first about the data themselves, not just to record like audio files and then store them somewhere. It's really important to start asking, like, what will we do with this acoustic data? Like, how do I format the metadata so that it's discoverable and readable for machines, you know? So that's to me, like, the data and what do we do with the data and how we format the data is the biggest challenge for AI methods. The rest is still in development. Basically, we're starting with bioacoustic AI. So we draw a lot on the computer vision literature. So we draw, like, bioacoustic AI is basically a lot of image recognition as well. But again, the challenge for all this AI is like the data themselves. The tool that we have now, we're able to classify species. We're able to detect, for instance, bird phenology. So what we do with TabMon. So we can already do inference on some biodiversity variables and across really different taxa. But we're starting here. So there's things that for now we can't do, or that's difficult to do. For instance, inferring species abundance it's still really hard. For instance, you can deploy an acoustic recorder somewhere. You can try to listen to bird vocalization. It's really hard to derive species. The abundance of a certain species, because one bird can sing more than others, etc. So it's hard. That being said, for some taxa, it has been done. It's possible to detect, like, individuals. Yeah, so we need to know what are the limits and where do the development need to go. Yeah, like, one of the major challenge to me is not the AI themselves, but again, the data, and especially the data storage. Like, we usually, as ecologists, we usually tend to just keep our data because we never know, like, maybe we'll use it later or not. So it's fine when we do that with Excel tables, but it's not fine when we do that with thousands of pictures or like millions of pictures and millions of acoustic files. Like, Just for reference, a simple acoustic file, like one of the, MP3 file that we record in Tabmon project is like 4.7 megabytes. It doesn't look really much, but when you do that over years and when you record 24/7, that start to become really heavy. It's like we're talking about like hundreds of terabytes of data. And so that can quickly be a problem, because where do we store the data? Like we can ask our institution, but some institutions don't have the infrastructure for it. So should the institution invest in such infrastructure? Well, it depends. Like in my personal opinion, is like we really need to rely on external partners, like on external class storage, etc. Like for instance, at NINA, we have private institutes. So like at the end of the project, the data can't be stored, or at least we don't have more money for data storage. So that's really a limiting factor. Maybe at universities, you can find some storage, but it's not scalable over the years. It's like maybe you will be able to store the data for 5-10 years, but then when the data set grow bigger and bigger, like at some point, someone has to pay for it in a way. So there's really a question on how do we handle this much data, basically. Like for now, the solution that we have is like store it on magnetic tape and have it in the basement on cold storage. And then like when we need it, we can retrieve it, but it's pretty slow. So I think in the ecological community, we really need to ask ourselves, like, how do we tackle these sort of things? Like, I think in the future, we'll really need to have the help of computer scientists or more technological people, etc., for kind of handling this kind of problem. But so far, we don't really have an answer. And Alia was mentioning, is like, it's not just ecologists who have this problem. It's like in astrophysics, for instance, it's like images of black holes and all that. It takes huge amount of storage also. So like we always think that our discipline is more important than others, but it's like the problem will be that all the discipline will want to like keep their data set on university server or somewhere just available, because in case we want to do something, but it won't be possible in the future.
Want to add anything? We move to the next.
Yes,
Yeah,
and I'd like to add something
yeah.
because you were asking about the challenges of doing this kind of analysis for the different taxa. Actually, there are important differences. We have very unequal performance of our AI tools in identifying, for instance, birds. We're most advanced with birds. We have AI tools, most notably BirdNet, everyone is talking about it, that can actually detect and identify birds automatically in sound recordings, but there are other fields and taxa, so groups of animals where identification is much more difficult. For instance, in the aquatic realm, colleagues of the consortium like Audrey Luby and colleagues have reported that around 700 species of aquatic animals have documented sounds. But based on what is a reasonable extrapolation of their data, they estimated that there are more than 20,000 species that are producing sounds underwater, and these haven't been documented yet. So you see that there are really very different challenges across taxa to do passive acoustic monitoring and to get to the identity of the species.
Yeah, and in addition, actually, like there's a lot of models, well, like you talk about a lot of models being developed, etc. Though I don't think that's a problem. There was the presentation before who the person was saying like it's better to have a local model than a global one, and I kind of agree with that. But what we need more is like to have some kind of this global model to build on top of them in a way. So I think one of the, is not really an issue, but for some is an issue, is like to have open source models. So I think that's like a really, really big responsibility for ecologists in general to put their model on like open source platform, because this will help inference. This will help everyone. Yeah.
And related to that, also I want to ask, even the performance of the same model in different backgrounds noise can do it different ways. So this is a thing that we are seeing, the same model with the same species in different environments with different background background noise, it can works that good, or it can works bad. So it means that in some way we will need to test the models that even we will do our models, or we use the already in our situation, because the same models can work depending on the background noise. How this is a key point also, that we don't really know how to manage to that.
Yeah.
And sorry. No, if I can add something, in terms of bats, for example, there are some bat species that they overlap completely. So even the most experts cannot distinguish between them. And I think that maybe the approaches that we are taking for modeling might be a little bit wrong. So we can still change the first approach and on our modeling. So in the past decade, we've been using random forests and different neural networks, and now it's AI. But the fact is that if these two different species cross in the landscape, they recognize themselves in the first millisecond. So we should be able to distinguish them, but we still don't know which is the right model. And there are a few researchers that have started trying different things, completely different approaches, trying to maybe find the right variables, the right models to do this separation. Like maybe tone of voice or something, different modelings, not just the frequency, which is basically the best measure that we use to separate bad species. So there's still a lot to go there and a lot to learn.
Yeah, that's also raised the question of model confidence. Like, can we really trust the AI or not? So like, there's no real answer here. Like, I think AI tools is, all AI tools is basically a complement of other kind of monitoring strategies. Like, we have the same exact problem with camera trap. It's like all the AI tools used in camera trap is the same. It gives a probability, and we assume that the probability is right. But it's like somehow if the model like classify a wolf as a dog or something, then we already missing a rare species, and then that can have like big consequence for the management. So it's like the problem of model confidence is also a big problem that we're trying to not solve, but find some strategies to account for. So there's some development in more statistical models, especially with data integration, like integrating data from AI with citizen science, etc., like to derive some true occurrence of the species. But I don't think we'll ever replace human intervention in the monitoring protocol.
It's great because you give me the floor to talk about how all this data, the sounds that how we communicate the results of bioacoustics, we cannot show to the stakeholders, to the spectrograms, all this kind of EA random photo, all these topics. We need to, as we want all this data from biodiversity monitoring, impacts on policy, impacts on decision making, impacts on creating EBBs. So we need to look which are the opportunities of passive acoustic monitoring to complement the ongoing monitoring, to give information on of some species, but perhaps some species that if we don't have passive acoustic monitoring, we're not able to use. We all know that passive acoustic monitoring has some advantages. We can have continuous monitoring. If we go one woman in a field, they can spend not 24 hours just listening and counting birds. We can let them working. They are not invasive. They can. So there is a lot of advantage, but there is problems. And how one of the things that we are still, I think, in the baseline in some, but perhaps in the group of bats that you, Adria, has been working, because you cannot go into the fields with the binoculars or something and looking for bats because it's difficult. So you have more of experiences of how to bridge the gap between researchers, look at the citizen science, decision makings, and how can we better translate this bioacoustic data into tangible outcomes of ensure they understood, and at the end, giving valuable information to take decisions. So what is your experience on, for example, in the bat groups, that it's one of the terrestrial groups that you have more experience on that?
Yeah. So communication, it's key. We all know that. And probably all of us had included in our project proposal this sentence saying that we will organize meetings for stakeholders and that we will have great impact on community because we will affect land managers and policymakers, and we have all written that, right? But the thing and the truth, and I think it's a universal truth, is that we all speak different languages, and especially scientists and researchers and policymakers, really different languages. And this is something, is a barrier that we need to break and we need to communicate. And this is something that within the BAD world, we have been trying in the last decades, specifically from the institution I work on. I think I came up here with 3-4 different examples, just to illustrate a little bit the challenges that we have to overcome over the last years, and also to bring some real examples, just to bring our foot on the floor. And, you know, I really like Benjamin's last intervention. He said, like, human intervention will not be replaced by AI or something like that. I totally agree with this. I'm very aligned with this opinion. And the fact is that humans can perceive a lot more. And the expert opinion and the expert knowledge is invaluable here. I've attended the talks this morning. I really enjoyed them all. But I've seen that all of them is like GB, 3 million points, and programs, and so many holistic projects with a lot of different taxa combined in one single repository, and everything like together. Nice, I love it. And I really like grouping things, compiling things. But if we lose the first stage where experts work on the ground with real data, actual efficient intervention never occur. Because the more holistic we go, the less detail we can get. And on the other way, it works both ways. If we only focused on the local scale with very useful and efficient intervention, we will lose the big picture. So we need both things. I'm not saying that one is better than the other, but we should never forget the human phase of passive acoustic monitoring in that case, or AI. First example, very, very easy. You know, wind farms have been implemented in the south of Europe a lot, well, also in the north, but here it's a massive problem for bats. We have these migratory bats colliding with the turbines very, very often, big mortalities. But then there is this AI, environmental impact assessment, and they do not know how much activity is too much activity to stop the turbines. So people go into the field, set up detectors, get the data. Okay, there are 500 bat passes. Great. Is it a lot? Is it not a lot? Should we stop the turbines or not? And it's really difficult to do that. So what we have been done for the last years is working on the ground, collecting acoustic data, terabytes of acoustic data in different seasons, different places, different environment and landscapes, and then draw this line in which we can actually say to the companies, above this level, you should stop terminally the turbines. This is something they understand. It's just one number with a clear idea behind it. And this is very efficient for conservation. And this is like one example of how can we communicate with the companies, not with sonograms, not with modeling, not with AI things. It's just they want to know above which limit they should stop the turbines. And that's it. That's one example of how should we really communicate, like bring all this scientific data into a summary, very precise about what they need to know. Another example, we have like a research line, very, well, I will not say historical, but we've been working on it for many years on pest control in agricultural lands, in crops. Bats, just as a, between brackets information, bats control a lot of insect pests in agriculture, and they do it quite intensively. So when we go to the agricultural lands, farmers always ask us, like, do we have enough bats? And I can replicate the example. I can set up several detectors, came up with the number 300 bat passes. Okay, is it enough to control the pest? It is not. Should I put bat boxes to attract the bats to the farm? Again, we need these thresholds. We need comparabilities. And this is the numbers and the situations they want to know. And this can be even translated to a more complex idea, which is indicators. We work on indicators with PAM, with passive acoustic monitoring. We calibrate our data, and we create this kind of gradient and scales in which anyone can set up a detector and know if the activity of bats is above the normal, below the normal, is like in the top 10 habitats in the country, or it's just bullshit, like no bats. You know, we have these indicators and multiple indicators that they can use it. The same for policymakers, politicians, and people working in the government. If they want to know, just put a clear example. They have 50,000 euros to invest in one single cave to protect it. Which is the best cave? Nobody knows. Should it be my criteria or my colleague criteria? Or the volunteers or the local people living next to the cave? So we standardize that. We create indicators, and then we sort out, we create this list, and we plot it in a map. And the map is what they really like, because they can use it. They can visualize very easy with several platforms on internet. They can choose and they can select where to invest the money. But this needs a lot of time and a lot of effort. So if we work on a very big scale, there's no way we can do that. So there should be people working on a big scale and people working into the translation at local scale. And the very last thing, if I have time, just to finalize. So we've been working in this platform like batmonitoring.org, which is basically a repository for bat data, acoustic data, based on citizen science. And we were discussing with some of you during the coffee break, okay, do we have volunteers or not? And how easy is to get volunteers? Volunteers means normal people, like normal people outside this room, like people that do not know a lot about bats, you know? And then how we engage with them? The only way we can engage with them is, for example, creating games. In this repository, we have create games to detect bats. And they know like the rankings of volunteers, and they have these dynamic maps that they can look after, and they have this sorting information about their locations. So it's kind of creating interactive things and indicators with colors, not numbers or p-values. And by doing this, our community has grown considerably in the last years. And I'm happy to say it's working. Every time it's bigger and bigger, and we're now running into these problems about, okay, we need storage, we need a standardization, we need calibration. So that's the way I close. So everything is just in a circle, but we need to communicate.
Thank you very much, all three. I think there's a lot of interesting things that has been arising in the roundtable, but I would like to, we have still some time to give the floor to the audience. If you have some comments or point of view you want to point out. If not, I will ask another question to the panelists. Do you have any comment?
Hello. I'm just interested to know all the data that you collect, and you try to have it available for the researchers and for the projects that are involved. Can we maybe generate a bigger machine to actually have it not only on AI, but to collect it as a big data engine, so we have the channels of communication between us or the partners of Biodiversa Plus to share it and actually access it, to use it?
I'm not sure I heard really well though. If you could just repeat that.
Yes, you talked about AI.
Yeah.
But AI is actually gathering information from everywhere. Am I right?
Just from the data you train the AI with. Yeah.
Yes, but we need to have not only valid real data, we need also to have live data accessing by all the Biodiversa Plus partners. Maybe it would be a really good idea to gather everything, not to use AI as a technology, but to use a wider environment, let's say, for just the researchers to know that everything that we collect can be used specifically for our projects. We can retrieve data and access it so that we can use it in our projects.
Yeah, like that. Yeah, I think that boils down to what are we collecting the data for? Also, like, you mean like it would be nice to redirect the data to a certain project if the research question align with it, for instance. Yeah, that would be really great. I don't think it's possible to. I mean, I think it's possible to create an AI or something just to like a ChatGPT like kind of. For instance, like I want this kind of data. Like, do we know where it is? But I think it's kind of overkill to train an AI for that. I think the important is like kind of what Kevin did with the worldwide soundscape is to basically have a really global database where we can basically find the metadata or find the contact person to contact the person. Because again, like the problem with acoustic data is not possible. I mean, it's hardly possible to store it completely available somewhere. So people need to choose the data they want to retrieve. So I think the worldwide soundscape is like one of the best example of this kind of thing.
Thanks. Maybe I can add something to it. So yes, indeed, we have started this project like 3 years ago. And it actually comprises more than 350 collaborators across both terrestrial and aquatic biomes. And so what we have created so far is a metadata base just to find out where, when, what, who recorded, as a first step to go towards a kind of portal or a platform where we could access these data. The problem is that the data themselves, we don't have infrastructure or storage to put them on, but we've made the first step in that direction. It's extremely costly to make these data available on hot storage. What is happening in most cases is that after a project, the data are written to an external hard drive or at best on some magnetic tape and left in a drawer or in some data silo. And so making these data available to everyone in hot storage is not only costly, but also emits a lot of CO2 because of all the energy that is used for it. So we have to be very careful. And the approach we have been using so far is that this worldwide soundscapes indexes and references where the data sets can be found so that in a directed manner, people can ask the people who own the data to retrieve them. But in a second step, what we will do is that we will ask our partners to install a kind of management system on which the project is currently hosted, and that they can upload their recordings there. And that in a distributed fashion, when all of these systems are going to work together, the strategy is really to go for distributed storage, that we can, in an interoperable manner, share these recordings so that they're not just usable to train AI models, which of course is very much in the focus and often the key to unlock the knowledge that is inside these recordings, but also to use the recordings for other aims.
Thank you. We don't have more time. I would like to thank Adrià, Benjamin, and Kevin to all these inspiring comments. As a moderator, I hope that if you are not familiarized with passive acoustic monitoring, now when you go out, you understand a bit more what is about all these topics. And I would like to make two small announcements. The first one is that Thank you. Just that will be at the end of this session in the classroom 19, that I'm not sure what it is, somewhere in that. The telephone told me that it's not that way. We will have a discussion session with the MARCO-BOLO representative member of the project, Pedro Junger, about the eDNA-based marine biodiversity reporting. So you are welcome to go there. And the ones that want to remain here, we will have now on stage Maria Santos from the University of Zurich with the OBSGESSION project, and she will make a demonstrative case of the project. Thank you very much for your attention.
Okay, so now I'll give the floor to Maria Santos to start her demonstration case.
Okay, good afternoon. So I took something from the previous presentation. Maps is what they really like. So let's explore some of the maps that we can do with old technology and new and upcoming technologies. So I have already introduced myself. I'm a professor in the remote sensing laboratories at the University of Zurich. And in this talk, I have contributions from many of the different authors because I expanded it a little bit instead of being just a demonstration case on the spectroscopy for biodiversity, but actually what can we learn from remote sensing technologies, especially in the optical domain, for monitoring and understanding biodiversity processes. Okay. So remote sensing technology for observation of the Earth is a 50+ years old type of technology, especially in the optical domain. What we have understood of how remote sensing actually works when we're thinking about biodiversity, and in particular with plants, is defined what we call an optical type. What is this optical type? Basically, what we have is light that interacts with plants, different molecules, different structures, different morphological characteristics, biochemistry, and that gives us an optical type. And this is the measurement that we get to be able to use to understand what plant is it, what its function is, which kind of ecosystems we have, and their extent. We have seen in the several last decades an evolution from what you see here, single band, early satellites that were collecting data at a global scale on a single band, being it red, green, blue, and so on and so forth. We moved into multispectral, so here we have different bands collected by the same sensor. And later on in this presentation, I'll present to you what is the novel technology on the imaging spectroscopy, which covers over 200 bands, and it will and is already deployed in satellites, but we will talk a little bit about that. What can we learn from that for biodiversity? What have we learned so far? Well, we have a wealth of Earth observation platforms. Here's two pictures, the top one from ESA, the bottom one from NASA, of all the sensors that are currently and ongoing observing different properties of our Earth system, the materials we want to understand, and the biodiversity that lives on it. We have been able to provide detailed monitoring of ecosystems so far, some plant communities, and even some species. We have been looking into ecosystem functional relationships among plant traits, for example, providing information on leaf mass area or leaf area index for vegetation and carbon models and other types of large models and dynamic vegetation models. We have been able to understand physiological processes such as photosynthesis, transpiration and respiration, and stress detection. We have been looking into plant and soil biophysical characteristics that are important for nutritional quality, water availability, and so on and so forth. And we have a lot of auxiliary data that enables us to actually understand biodiversity, its processes, and the biosphere as a whole. But we have seen a story of some on the left. On your left-hand side, you see, for example, Landsat acquisitions. The top left is 1984 until 2012, and so on and so forth. So with these repeated technologies, we've been able to monitoring over time what happens to any place on Earth that is covered by this technology, of course, with some limitations we will cover later. Moreover, we have seen an advancement on how we actually perceive the spectral properties that we can retrieve from satellites, airborne or handheld sensors. Because one of the emerging hypotheses from ecology is that there is a variability in the spectral properties, and this might be related to some sort of ecosystem heterogeneity or even the plant community composition that we see at certain locations. And given this assumption or hypothesis, then you can already make the math. If we associate spectral diversity to number of species or composition, then we can calculate things like local richness or turnover in species composition, or just create nice, colorful pictures of biodiversity that we don't yet understand. But we can actually produce some more informative pictures. This just came out of the press from my lab. This is the Amazon with Sentinel-2. We're processing actually the spectral richness over the entire extent of the Amazon. And so what we can see here is that it reproduces some patterns of primary and secondary forests, deforestation for the Amazon. And we're now trying to understand what do these different richness, so how many spectral profiles that are different within a given location relate or not with the composition in the Amazon. We can also make more colorful maps. And here what we are displaying is how similar or dissimilar are the spectral properties within a cluster of pixels. We use typical ecological metrics such as turnover, and we calculate what we call beta diversity for spectral reflectance signatures. And what you see here in red are spectral signatures that are more dislike the ones in their proximity. The ones in blue are spectral signatures that are more alike the ones in their proximity. So with that, we can start to understand a little bit what is the heterogeneity and how is it changing spatially. And in the future, when the lab continues doing this, we might be able to also have this turnover in time as well. For remote sensing, we've also understood so far that it's not just about mapping the number of species or how do they change in their spatial domain, but also which kind of functions these species provide. A lot of remote sensing is used for plants. And then, of course, we want to understand how plants function because they provide us a lot of information on ecosystem functioning and so on and so forth. So how does it work? Well, I've already showed you that we work with what we call these optical types. Well, the optical type of a plant is represented here on the right-hand side. And what you might be able to read on the top is that different parts of this spectrum, the absorption features, relate to different chemical components and the different biochemistry and physiology of the plant. So because of these, we are able to actually start to understand how plants function and other processes at local, but also at global scale. Many of you may be familiar with NDVI. Here's the original publication in May 1978. And so Compton Tucker, he was very clever. He just looked at, oh, look, plants absorb in the red and they reflect a lot in the near infrared. So if we calculate a normalized difference indicator just between these two bands, we can produce, and here's the first map of NDVI at a global scale. So this was produced by the NOAA weather satellite. And you can already see, maybe if you're familiar with global distribution of different types of biomes, there is quite a bit of patterns of what we see in these maps, even with very simple relationships between what remote sensing optical types tell us about plants. We have seen a lot of advancements. We have seen several papers trying to produce different types of trait maps at a global scale. There's one upcoming, also AI-based, about 35 plant traits being produced by all the satellite archives that we have currently. And then we can use these to actually understand some ecological processes. You might be familiar with the graph here on the left middle-ish, which depicts the plant kind of functional strategies. And the one on the right is what we can retrieve now with remote sensing. And you can see that they are remarkably similar when we use in situ data with that we retrieve from in situ and remote sensing combined data. And then you might be thinking, but then what do we use this for? Well, we use it to understand certain types of questions that we are curious about in ecology, being it for monitoring or for understanding processes and responses in the long term. So for example, we can use these maps of traits to look into phylogenetic relations in major lineages. And one of the options that we say is that, okay, so plants that are phylogenetically related should have a spectral signature that is more similar than plants that are not phylogenetically related. And so if we have maps of traits, we can start to depict what kind of phylogenetic relationships might emerge there. But we might also look into or try to attempt to get to some information on genetic diversity. This is a group also from the remote sensing laboratories that have this new project on genes from space. And here they make some very simple assumptions based on metapopulation structure and habitat and number of effective populations within the habitat of a given area. They use typical remote sensing classification, extent of forest or different ecosystems, and they make a prediction using GBIF data when the species are present and absent to calculate effective population sizes and showing differences in habitat types as well. So with that, I've showed you what we can do with multispectral multispectral remote sensing data. But the topic of today's demonstration is what can we do then to better understand biodiversity and its processes, given these emerging technologies called imaging spectroscopy. So when I started working with imaging spectroscopy some time ago, I will not age myself, we were hired to actually detect invasive species with imaging spectroscopy. So one of the premises with having this very finely resolved spectral response is that different species will have different spectral signatures, and therefore we should be able to detect at least some of them. Invasive species are a great example for that because they tend to either be slightly different in their phenology or in their structure or in their physiology. That's why they are invasive and successful. So therefore this technology was used, and we used many different types of data. So HiMAP and AVRIS are the airborne, ASD are the field measurements that we took, and we collected in situ data on many different species, native and non-native, to compare them. So then we run into a PCA to see, okay, can we differentiate these invasive species based on this information for different types of groups? So you see there the types of groups that we see: dry grasses, grasses, aquatic macrophytes, cane grass, shrubs, emergent, floating. So the groups come out very nicely when you put them all together. And then what we looked into was, are there certain absorption features within the spectral response that are different between native and invasive alien species? And what you see on the right-hand side graph, the red are the ones where the invasive species have higher reflectance on those regions of the electromagnetic spectrum. The green are the ones where the native species have higher reflectances. And based on this information, we are able to, at least some of the species, to identify them and to inform the managers from these many different locations in the United States that asked us to produce this type of information. What we have seen also using imaging spectroscopy is for understanding richness and turnover. I gave you some examples using Sentinel, so multispectral data, but actually the original research was using the Carnegie Airborne experimental airborne facility over some parts of the tropical landscapes to actually derive this idea of how can we actually calculate richness and turnover using spectroscopy data. So here's the original data. So you see here the flight lines from the airborne platform, and here you have a measure of richness and a measure of turnover. And you might be curious, how does this work? Well, actually what the method does is to calculate clusters of spectral profiles and then classifies each one of them pixels based on these different clusters of spectral profiles. And like many types of analysis that we do in ecology, that we take the first PCAs and they explain most variability, when we're talking about mapping richness, we want as many PCAs as possible because some species might be very rare, so therefore not captured in the first PCAs. And then we predict what kind of spectral clusters would the pixels be located, and then we count how many pixels we have in each one of the clusters, and that gives us our idea of the richness. Since then, we've also seen the emergence of what we call spectronomics, so understanding how these biochemical and physiological properties of plants can be extracted from spectroscopy data, being it in the field or airborne or in now existing satellite platforms. So what this means is that we can actually use the information that we retrieve from the spectra to derive, for example, functionality. So some of the first papers came already about 10 years ago that predicted using partial least squares regression and other statistical methods, what would be the composition and the content of different types of traits as using imaging spectroscopy data. We have seen very recently using AIs and different types of models to see how the predictions compare and contrast depending on the type of model that we use. And so this is a very active area of research of can we use this type of information to understand plant traits and therefore their functionality. We can also do this in the aquatic environment for those of you that are interested in that. So this is we can use, for example, vegetation indices that relate these areas of the spectrum or these areas of the spectrum or these areas of the spectrum. They tell us different things. So for example, the ones on the top, they tell us about chlorophyll and other pigments. The ones here highlighted in yellow, they relate to the leaf area and morphology of the plants. The one here, they talk about leaf water content, and the ones in the bottom, they relate to lignin, cellulose, and salinity. So you see that the different parts of the spectrum actually can tell us a lot about how the plants actually function and what makes a plant a different species from the other one. And then we can use this information, for example, to look into whether the plant traits change over time. So this is based on a time series of 10 years and in 2000 and 2010s, and the climatic conditions changed. So we wanted to see if the plants were actually responding by changing their trait functional response based on imaging spectroscopy collected every year. But there's more. So I talked about genetic diversity in terms of retrieving it based on some sort of relationships of effective population size. But actually what Eva looked into was she actually created a database of beach genotypes, so Fagus sylvatica, and then she also measured their spectroscopy using airborne imaging spectroscopy. And here's what you see here, whether different components of the genetic composition of beech trees looked different across the season, and whether we can retrieve that with the imaging spectroscopy. But then, of course, there's some uncertainty that needs to be accounted for, because when we're talking about making maps that everyone likes, we also need to say how robust are our maps. So that is one of the things that there is more, but there's more also in terms of the biomass. So within OBSGESSION, we are monitoring the Swiss National Park here because we want to understand biomass and the new herbivore populations that exist within the park. So we have been monitoring this with a prior sensor called Airborne Prism Experiment that we collected data from 2010 until 2019. Over the Swiss National Park, you see here the map on the right, the flight lines that were collected over time. And then we wanted to assess what is the effect of certain types of data pre-processing that we do for remote sensing data on the accuracy of the models, in this case, fresh biomass. So we see here that the best atmospheric correction model that we can use does quite well across different phenological states. So, and then what we can also see is that depending on the time of the year and the type of pre-processing, we might see different influences of parts of the spectrum. So why imaging spectroscopy? Well, we spent quite a bit of time developing what we call airborne sensors, and I would like to introduce you AVRIS4, which is part of a sequence of airborne sensors that have been developed first by the Jet Propulsion Lab at NASA, and so on, and then they got also developed at different companies, and now, for example, Planet also have such type of sensor mounted in small satellites and in different types of architectures and number of satellites. So why AVRIS4? So AVRIS4 is a sensor that builds on the previous sensor that was built in the remote sensing laboratories in Zurich, but also in collaboration with JPL, we kind of merged the two types of sensors development to develop what is currently one of the best airborne sensors that we can have with this technology. So this is emerging as a collaboration by the University of Zurich, the University of Applied Science in Zurich, and EPFL as well. And so what is it? So as I was mentioning, it is to maintain these long for 3 decades airborne platforms that have informed what we can do about with imaging spectroscopy, not just for biodiversity, but for different types of applications. So we have AVRIS Classic. I showed you some examples with the invasive species. That was with AVRIS Classic. We have AVRIS NG. We had some acquisitions in Europe and also in many parts of the world, which I will show in a moment. NASA has just produced AVRIS 3, and Zurich, in collaboration with NASA, produced now AVRIS 4. So what are the technical details and why is it so special right now? Well, we've talked about imaging spectroscopy in the hyperspectral domain can collect over 200 bands. So if we can calculate NDVI with 2 bands, the 200 bands give you really this very detailed spectral signature that is very important for different types of species and different types of biodiversity applications. We have a line scanner that collects about 1,280 pixels on an image. And we can also get to very high spatial resolutions. Right now, our minimum spatial resolution is 30 centimeters on the ground. So you can imagine that because we have this airborne platform, we can actually resolve very well spatially. So how does it work? You might be curious. So basically, every time that we get a request for an airborne campaign, we have to check the weather, we have to plan the flight lines that depend also on the position and in the location and the extent. And then we ask also the researchers what kind of spatial resolution would they like for the target that they are interested, what kind of overlap, because sometimes people want to have overlap in the flight lines, and what kind of temporal constraints are there for the region because of the different solar angles. And then the plane is piloted by 2 pilots, and there's scientific staff that goes along when the sensor is being piloted. This is not unique for UZH. The NASA campaigns are also like that. So we have quite a lot of personnel that is required to maintain this type of technologies. And then if you were to use this data, what is it that you get in the end if you want to do such acquisition like the ones we are doing for OBSGESSION? Well, what the team of AVRIS 4 actually does is all of these 4 steps that are here. So first, we do some meta-analysis, file sorting, file renaming, and then we have radiometric calibration to calculate radiances, and then we do geometric processing, so how to account for airplane movement, yaw, pitch, and roll, and the camera properties, and also to localize the pixels in the 3-dimensional space that the airborne sensor collects. And then finally, we go to the surface reflectance retrieval, which is this nice spectral signature that I was showing you that we then use for the biodiversity products. So then, how does this all look like? Well, here's an acquisition over Legern in Switzerland from the 21st of September of last year at 0.7 meter ground surface resolution. So each pixel in this image is 70 centimeters on each side. And so if you talk about data storage, we can talk more. So here's just to give you some examples of some visualization. So the top one is the typical RGB image that we can combine, but because we have 200 plus bands, we can make many of these combinations. So this is the common color infrared that tells us like vegetative areas. And here's the SWIR, so a shortwave infrared combination that tells us other properties about the vegetation. So if you're interested also on how the final surface reflectance products looks like, this is the geometric quality. So you see that the pixels are very highly positioned. So that's why we ensure up to 30 centimeter spatial resolution. And you can see here that it took a bit of time for the geometric position to be good in a Swiss standards. And especially when we're talking about highly topographically variable areas, the Swiss National Park took much longer to get the geometric position well, so that our 30 to 1 meter pixels are very specifically located. And then this is how, for example, at 40 centimeters resolution, how the final reflectance product actually looks like. If you're more technical and you like to see spectra, this is how they look like. The red one is the one collected by the airborne sensor. The blue one is the one collected in the field. So you see that there's very high match between what we can measure in the field and what we can measure with the airborne sensor as well. And you might be interested in application examples. I already showed you what we have been doing with previous sensors. And so what can we do with this sensor and also the upcoming sensors that we expect NASA and ESA to launch in the coming 5 years or so. So for example, if you're interested in plant functioning, so the top one is a chlorophyll carotenoid index. So that tells you more or less what kind of content of chlorophyll, so stress detection on plants in Lager in this case. You might be interested how this red edge that tells us like whether the plant is starting senescing or not behave, and that's what the middle one tells us. And then the normalized difference water index is self-explanatory, tells us about the water content in the canopy. So this is just pure playing with mathematical indices with some of the specific absorption features within the data set. And then we can also think about other types of combination. For example, the bottom one. So here is the typical RGB. So sorry about that. We want to go back. Yeah. So this is in our eyes, the visible type of image that we will obtain. But then if we combine all of those previous vegetation indices as indicators of plant functioning, you can see that the combination of the different colors tells us what is the diversity of functions that we have, even within a beech forest that is relatively monotonic in Lagan forest. And then for OBSGESSION, this is the collection over Lagarde. So you see on the left-hand side the original spectra, and then we see here after the pre-process, all the processing steps, what we actually obtained from this sensor. And I don't know why this is being automatic. And then what we did for OBSGESSION was actually to collect leaves from the top of the canopy of the trees so that we can get their reflectance spectra with the handheld spectrometer to make the correlation between the in situ and the airborne platform. So this is just a picture of our climbers, because fieldwork is also very fun when we have very tall trees. And then if you're interested in other types of applications, we've been working with EMPA in Switzerland to detect methane plumes. So this type of sensor actually tells us something about methane being emitted, in this case from an human infrastructure. But if you're interested in wetlands, this might be also a sensor that can be useful when we're thinking about methane. And so here you can see a map on the stratification of methane in the atmosphere that can be retrieved with this sensor. And you can see over different locations that you might be interested, how these types of gases get distributed in the atmosphere and might interact with what you're interested in. And then if you're also interested in more extreme environments, for example, glaciers and biodiversity in these very extreme environments, the sensor is also very good to investigate what kind of debris cover exists in these areas. And because of the high spatial resolution, might be able to tell us something about colonization processes in retreating glaciers. And you might be thinking, okay, what data is there out there? Well, up to now, most data is made available by NASA JPL. So for the Average Classic Data Portal is depicted here. So these are all the data sets that have been collected over the last 25 years. They have been reprocessed and reanalyzed. Anyone is free to download them and analyze them. Average Next Generation Data Portal, we start to see the European campaigns if you're interested, but there was a campaign in India, in Greenland, and in Alaska. So many types of environments have been sampled with this type of technology. And then we have our own ARES. So this is the campaign that occurred in 2021 with the average NG across Europe. And we have a new portal that we're now experimenting with the new acquisitions that we're making since 2021. So you think, well, we're all resolved, we're all good, but we still need to learn a lot of things. So This technology is becoming more mature, but there's only restricted groups of people that have been using it in a more systematic way. And we've talked a little bit about uncertainty, and when we're thinking about biodiversity monitoring, we need to be really careful about uncertainty, especially if you're looking for very specific spectral absorption features that might be just a tiny little bit different. We want to make sure that what we're measuring is actually the signal and not because of uncertainty. So what we've been looking into is how can we actually estimate the uncertainty at the sensor so that when we deliver these data products, we actually can assess something about biodiversity. So what we do is to develop physical models that tells us for every specific waveband what is the measurement that should be retrieved and what the sensor retrieves and what is the uncertainty band that we have at each wavelength. If you're more spatial like myself, so what it can produce, this is for a satellite, DESIS, but this is what we aim to produce also for the airborne. So this is the uncertainty map at 630 nanometers. So if you're interested, for example, in sun-induced fluorescence or any type of other fluorescence, maybe you want to know what is the uncertainty in your spatial domain. If you're interested in how your different plants might play a role or whether the sensor is able to capture the information for your specific area, you're able to produce these spatial uncertainties. And within OBSGESSION, what we're doing is we're examining 3 types of biodiversity products that are commonly used. One is functional diversity and spectral diversity. So those are retrieved directly from the remote sensing signal. And then the 2 other types are species distribution models and dynamic vegetation models, which use remote sensing inputs into these models. And we want to see how these uncertainties from the sensor propagate into the outputs that these models actually produce. So what else do we need to learn? We see a lot of data coming in. These are the Copernicus Sentinel expansion missions. You see here the imaging spectroscopy mission CHIME, but we see synthetic aperture radar, so ROSE-L. We see different types of microwave and different types of land surface missions coming up soon. So we will see a lot of expansion of these missions. So we also need to assess how do we actually integrate these data. So here's just a picture just for fun from LiDAR of how we actually can assess forest structure and carbon. LiDAR will be coming up and SAR will be coming up in new upcoming missions. So if we drape the optical information on top of the LiDAR, then we can start to see the different traits in the 3-dimensional structure. We might want to look into SAR. It's very good for water properties as well. So the SAR enables us not just to detect, for example, motion infrastructure, but also water properties in the canopy. And it also gives us some sort of sense in 3-dimensional structures. So if we combine these with the possibilities of imaging spectroscopy, we start to have a much more complete picture of what happens in the canopy or in your ecosystem of preference. And then we see now also in the thermal domain, hyperspectral cameras as well. So we start to understand, for example, energy balancing in forest, or for example, thermal niches for non-plant organisms or other types of organisms that might be responding to changes of temperature within or outside of the canopy. But we've talked already about many new methods, so that's still a lot to learn. We talk about AI, but I would like to make a call for explanatory AI, that we actually understand what the AI is retrieving. So we have a lot of new methods emerging that enable us to get more out of existing data. For example, and this is a special example for Petteri in Finland, of what and how can we estimate biomass using these advanced methods and combination of data sets. So traditionally, we have in situ methods that enable us either through this destructive measurements or using allometric equations to estimate biomass. We can use remote sensing from airborne platforms like the one I've just showed you, or spaceborne platforms, but maybe we can and are combining the input information. Say you have an RGB image, you use typical statistical methods to obtain some prediction of biomass in a given location, or you have more coarse resolution that you want to upscale to obtain biomass maps. But now we have methods like guided super resolution, which use, for example, inputs of high resolution satellite image and low resolution, for example, above ground biomass maps and produce high resolution above ground maps. So these techniques enable us to actually combine the power of the many different types of data sets that are being produced. So with that, maybe you think, okay, but how well do they do? So for example, here, this is this is what is in a given location. So this is what we call the ground truth, is an observation of above ground biomass based on in situ observations. And then the models produce these different predictions. If you zoom in, you can compare the different predictions for the same area, and you see that some models don't do very well, and some models start to approximate very well what we would observe in the target image. And then we can calculate the difference map. And you see here that the guided super resolution models have a much lower color in the difference map, meaning that their predictions are matching very well what we want to achieve. And so you might be curious, but do the models actually do well across different biomass ranges? So this is what this graph here says. So on the right-hand side is a higher above ground biomass, and here for low biomass. So for low biomass, it does quite well. For higher biomass, not so well. So we need to start using these methods with some sort of thinking about what is it that we want to predict and how do we actually interpret them. So I started this career imaging imaging spectroscopy because at the time that was not possible. It's very nice to see that many years later we can understand quite a bit, and we will be able to have everyone globally actually using this type of technology to understand better and understand what we can do and what our pitfalls are. So with that, thank you very much for your attention. And by the way, that was a real image that we acquired last summer with the heart-shaped clouds, which we don't like clouds anymore. I don't know if there's time for questions or.
So if anyone has a question, yeah, we have some time for questions. Yeah.
Thank you. It was an amazing talk. Maybe I understood, I don't know how much of it, but some parts I understood, especially being trained as vegetation ecologist. To me, yeah, usually what I do is go to the field, make quadrats, imagine your grids, and I count species, all the species in the quadrat. So I'm wondering how well does correlate, because you were explaining about measuring species richness based on spectral variability. So I wonder whether there are studies which already correlated the results from this variability with ground truth data and how well they perform. And especially I wonder about forests in which, sorry, how should I, is it okay like this? I don't know. Okay, and in forests, maybe I can. Okay. So in forests, you also have the herb layer, which is very rich.
Mm-hmm.
I mean species, but usually there cannot be observed when the forest is developed. So I assume they perform very badly in terms of like if you want to see whole layers.
Mm-hmm.
So yeah, just my question, like whether there are studies who correlated ground truth data with.
So as a good ecology, I'll say it depends. So what does it depend, the accuracy of these types of models? It depends if your canopy covers the entire pixel or if there's gaps. So if you see gaps, you see the effect of the understory in them. The second thing that it depends, and we're going to have a session in the Living Planet, Living Data conference later this year, is that a lot of the forest plots that we get from in situ don't match exactly what we need from a remote sensing. So we've been very opportunistic using all the data that exists, but should we design actually in situ observations that can be matched to remote sensing products? And then the understory, yes, it is a problem. Well, it is not a problem. Many of these satellites actually can see things from multiple angles. but often we correct it to what we call pseudo nadir or nadir. So actually, if you use the full information from the different angles, you might be able to detect some of the understory. And then, of course, you can make use of phenology in cases of deciduous forests where you could see the understory coming in earlier or later. For tropical forests, that would be very, very difficult. What we can do is to try to decompose the signature that we get at the pixel level by exemplary. So we use spectral libraries to decompose what could the signature be composed of. But it's a very challenging question, and we're just now starting to understand in which ecosystems it might work or not, because what we see is an heterogeneity on the spectral signatures. So that we can say. Whether it's attributed to the number of species or other characteristics, that's what we need to move on forward.
Hello, everyone. Okay, so we have a very packed table. So I'm going to start by semi-introducing the speakers. And I say semi because I just told them that it's way more interesting if they introduce themselves than me just reading the bio like I'm reading a LinkedIn page. So I'm just going to say the name and a little bit of what they do, and I'm going to leave them to highlight something about the work. So you're going to start the other way around. So Michael Cunliffe is the Director of Science and a group leader at the Marine Biological Association and Professor of Marine Microbiology at the University of Plymouth in the UK. Michael, do you want to say something over yourself?
Yes, I'm not allowed to reveal that I'm a tap dancer, so I was told that. Yeah, so obviously this is an eDNA panel. Just a bit about, I mentioned earlier, I've been working with molecular tools to study microbial diversity for nearly 20 years. And I guess because I've come from the microbial world, really the only way that we can study diversity really is to use eDNA. So for me, it's very natural and it's the right way to do it. And as I've evolved, I've got more interested in microbial eukaryotes and fungi and other things. And I've really appreciated some of the clashes now with some of these other groups that people think you can't use eDNA. Yes, that's possible.
Thank you, Michael, and welcome. Right next to Michael is Emily Boulanger, and she is joining us from the Ocean Biodiversity Information System. And she's a key member of the MARCO-BOLO project's Work Package 2, which is dedicated to validating the use of eDNA for marine biodiversity monitoring. And I have to say that I really like the name of MARCO-BOLO.
We didn't come up with it, but thank you. I'm Emily. I'm a marine ecologist, mostly working on fish biodiversity. And for me, eDNA is kind of this amazing sci-fi tool that gets us to kind of, monitor these aquatic ecosystems or marine ecosystems without seeing them. So I think it's really intriguing. And now since a year and a half, I work for OBS. So those that don't know it, is kind of like the ocean equivalent to GBIF that you all know. And we are involved in different eDNA projects, focusing a lot as well on getting the eDNA-based biodiversity data in these biodiversity platforms in a harmonized data manner and validating then also the use of eDNA in the framework of MARCO-BOLO.
Thank you, Emily. Welcome. And right next to Emily is Javier del Campo. He is a microbial ecologist based at the Institute of Biología Evolutiva, joint center between CSIC and the Universitat Pompeu Fabra here in Barcelona. And I have to say, I've been following you on Twitter, when Twitter was Twitter, for many, many years. This is a fun moment.
Thank you. As Michael, I have been working for, probably 20 years on metarecording because I come from the microbial side of things. I study coral symbionts, and in my free time, I manage a metarecording reference database, and I have been doing that for 10 years. So I know a lot about all the frustration that are linked to the analysis and meta-analysis of eDNA or /metarecording/microbiome data. And I'm here to try to help with this conversation.
Sounds like a fun hobby. And least but not last, Elaine van Ommen Kloeke. Did I say that correctly?
Close.
Okay, thank you. She's the project manager from Arise at Naturalis Biodiversity Center in the Netherlands and coordinating the connection with other infrastructures initiatives such as Biodiversity Genomics Europe, Biodiversity Meets Data, GBIF, eyeball, Biodiversa Plus. And do you want to add something else?
Hi. So I have, once upon a time, I was a biologist. No, I like the pragmatic approach. So I enjoy research because I come from the research world, but I also like how can we now implement this into products, something that's very useful, scalable, so we can actually do this.
Thank you. So we don't have a lot of time actually. And I think that as an introduction, I'm going to stay with what Emily said, that eDNA is this sci-fi tool, right? And I'm sure that a lot of why are we here? How can we use this sci-fi tool for biodiversity purposes is going to come up in our conversation. I'm just going to go straight to the question, so we have more time instead of just the 15 minutes that we were supposed to have. So, so I'm going to start with Javier. So you come from the field of microbial ecology, where genomic approaches have been used for a longer time than in other taxa. Could you share how eDNA started being used for microbial studies and what lessons this longer history offers for its application to other groups?
Okay, so yeah, we have been using, let's call them eDNA. For me, it's difficult to call them eDNA techniques because we never call them like that, but now we have to because the term has become more popular than the term we used before. So I'm gonna call them eDNA. But the method is the same one you will use for microbiome analysis. So it's exactly the same. So I commonly call it metabarcoding. So we have been using it since the 90s, not me, because I was still at high school, but these techniques, what they brought to the field, first of all, is that they changed completely our conception of microbial ecology. And they changed it for us studying the environment, but they changed it for also physicians studying humans. And they started to treat humans as an environment as well. And they adopted this ecological approach to study humans and to study and veterinarians to study animals and so on and so forth. So for us, that was a game changer. We suddenly could start doing ecology using this data that we couldn't do before because we were just isolating microorganisms from the environment. That was very limited. That was one of the first changes. And the second is all the diversity that we could unveil using these metabarcoding approaches. That changed completely the tree of life forever. And it's still nowadays changing the tree of life, especially for microbes. It has reshaped the whole tree. And that happened intensively during the first decade using eDNA methods. And then I must say that this method has certain limitations. And after this peak, now we are assuming and embracing all these limitations, and we are incorporating other strategies to study our systems that are not eDNA. I suppose that in this community, you are still going up in this slope, just reaching the peak, and then you will realize all the limitations that this sci-fi method has, and you will start adding other tools and combining this type of information with other type of information that for a period of time you might consider that is not as good anymore because you have eDNA, but all the information you can use, it's going to be useful, and combining different strategies is, in my opinion, the best approach, and embracing a systems approach to study your environment and also for biomonitoring, I think will be the wisest way to proceed based on the experience from the microbial world, I was saying. But Michael might have something to say about that as well. Some microbial guy.
You're on the spot, Michael. Do you want to add something?
Yeah, I mean, I think often we come to meetings like at the workshop and decided that somehow eDNA is this sort of panacea that's going to make everything come okay. And I think that's definitely not what we're saying. And as the same as Javier said, I think calling it eDNA does feel a bit strange. in theory, we could be in a place relatively soon where we can just, I mean, there is some philosophical thought process that we just sequence everything. So once we have the power and the capability and the storage and supercomputer, we could just sequence everything, and then that would be okay. We're not. And I just want to put a pin in the point of sometimes we have to sort of remind ourselves in Western Europe that we can just go and sequence stuff, and we've got money in our pockets to do it. And I am a bit nervous of that. That's not a global view of approaches to biodiversity assessment. And so we just need to have that in our heads.
Yeah.
We've got these like mega sequence projects. You know, there's a large proportion of the world that just can't do it.
Yeah. Yeah, I really like the fact that you just point out about the limitations, because usually we don't want to hear about limitations. We just want to hear about the possibilities, but you really need to contemplate them. And the limitations might be technological, might be economical, but might also be political. So, Elaine, you are with the ARISE project, which is a remarkable national scale initiative. So what are the main enablers that allow the Netherlands to move forward with such a comprehensive eDNA strategy? And what were the limitations, right, that you needed to overcome for the project to happen, basically? And are there any lessons that we could learn if we want to do the same in other countries?
Reference data. Yeah, you can't do environmental analysis if you don't have the references. So you first really need to ramp up your effort of knowing what species do I want to at least have in my reference database, have the references, make sure they're correct before you can do something else. But it's also being smart. Like I said, I like pragmatic approaches. If you're working with people in the field, you need to lower some boundaries. So we've created these little kits with instructions, and all the things are already inside. We created apps so they have a very clear way of, this is what I should do in the field, and this is how I make sure my data is captured appropriately. There's even instruction videos. It's as simple as can be, because otherwise you're not going to engage on a larger scale with people in the field. So these kind of boundaries really need to happen. But also, as Michael pointed out, you also need to convince people that this is the appropriate way forward. It's adding something to the mix. It's not replacing anything. And it's definitely not a God's gift answer to everything. You still need to have a human going to the field if you find something interesting. Yeah. You still need to have someone interpreting the data. Okay, AI will come along. But there's still a lot of things that you do not necessarily know, but it also creates opportunities to do way more than you could do before, or investigate species communities or species interactions with this kind of tool. So lots of boundaries to be taken, but also lots of opportunities that can enable such things.
And in the sense of boundaries and also possibilities. So as you said that in a past life you were a biologist, I was a marine biologist in the past. And I used to study marine sponges. I used to do genomics. So all of the things that you're saying right now, I'm having kind of like good memories and bad memories about sequencing sponges with all of those microbes inside, and definitely the limitations. So Emily, you work in the marine biodiversity space, and with this beautiful project, MARCO-BOLO. So what are the specific challenges and opportunities of applying eDNA to marine ecosystems? And also, if you compare it to terrestrial and freshwater, there's a lot of possibilities, but there's a lot of barriers as well.
For me, the first challenge is a technical one that comes to mind for me. It's that this marine space is this open, highly connected. So the oceans are so connected, but also very heterogeneous. And I'm coming from this macroecologic perspective where we're looking at the traces that species leave in their environment. And there, the transport of these traces play an important role in then how we're going to be able to interpret them and monitor the ecosystems. And so I think in the marine space, it's these currents are not just from upstream to downstream, but they're going to be in all different directions, in all different dimensions as well. So I think from a technical point of view, this is still, if we want to do very precise biomonitoring, kind of a technical hurdle to really understand the hydrodynamic system you're working in, so that you know, okay, the species, this fish I detected, it could come from a couple of 100 meters in that direction, but not that direction or that direction. So it's a personal thing, I think is quite important. At a larger scale, I guess the marine space has been kind of understudied or compared to these terrestrial or freshwater spaces because it's more difficult to access. You can't just walk out there and go see for yourself. You have to have these vessels that can bring you at sea. You have to have this equipment that can go underwater, these people that can dive. So that's a big challenge that means that the ocean has been understudied compared to these systems, but that's where also then eDNA can be this great opportunity, is that, as you said as well, like the sampling can be very easy. We can give people already kits that are all pre-prepared. So the sampling becomes very easy, affordable and accessible. And so we just need to take a couple of liters and we can do that in the deep sea where we can't send divers. We can do that in difficult to reach environments. So that's the big opportunity of eDNA. And then if people also put them in the biodiversity databases, then you can start filling out a lot of these big knowledge gaps that we have at the global scale.
Well, it's affordable in one sense, but very expensive in the other one, because you have to send people to the ocean and vessels and or drones, and it's one of the main limitations. And you said people as well, like sending people under the water to sample. I'm guessing that you're not thinking about analyzing anything under the water, right?
No, I meant that diving can be very expensive and difficult, and yeah.
So, and in this topic, which is like this of harmonizing across realms, and Michael, you have experience on that, right? Like,
Yeah.
So how realistic it is to have this like unified methodological framework for eDNA that can work in both marine and terrestrial environments, or not at all, and we need to approach them like they're just different frameworks to work in each of these spaces.
Yeah, so I haven't worked with terrestrial soils for quite some time. We've worked with sediments and seawater and sea ice, coastal water, open ocean, microbes in symbiosis, and I sort of don't think there will be a framework. Yeah, sorry. Yeah, so I've worked earlier on soils, but also sediments, seawater, sea ice, range of different systems, and I don't think we will have a single framework for them all. But even if you think about the issues, one of them is scale. So you take a gram of sediment, what's that equivalent to in terms of seawater? Is it equivalent to a liter of seawater? And then the other big one that I think we don't really think a lot about is time. So, and the importance of seasonality within these different systems. And so where we are in Plymouth, seasonality in the open coastal system versus seasonality on land is really quite different. And how do we capture time? I don't know. Also, I really like the comment about storming. It's really made me think hard about where we are at the moment. And one of the places that we have been over the last 2 decades is a lot of contention about which primers do I use to do my metabolite. This whole primer debate, and people very precious about their primers. So there is a move now to some what are described as universal primers. They're often called the Fermat primers. These ideas of PCR primers that will capture everything. But the only way to make that work is you have to have very deep sequencing to sort of make that work. And I don't think it's really dated. And then another group of organisms that I work on, not in MARCO-BOLO, but other fungi. And you can't do fungi with some primers and not others. So I'm not sure we will have a single unified system, personally.
Mm-hmm.
But I also not sure why we should.
No, it does make sense. Yeah. And so on the other side, we also have the harmonization across taxa, right? So Javier, So given your background, what are the key challenges when trying to harmonize microbial eDNA approaches with those used for larger taxa like animals or plants?
All the challenges. It's a big challenge. So we can start by the different definition of what a species will be, different groups. And if we start there, then that conversation is over, done. But also the different barcodes that you will use to study different communities. That's another one. Even in the microbial realm, for most of the microbial eukaryotes, we use certain primers, but for fungi, we use a different set of primers, basically defined by the resolution that those primers will have. And this resolution is defined by the knowledge we have as well. Okay, so the less you know about this group of organisms, the more universal and less specific can be those primers. That happens for bacteria as well, that you don't have all this, you don't have, in my case, I'm a protistologist, so I study microbial eukaryotes, so we have 300 years of biological knowledge on these organisms, so we have lots of taxonomy, etc. So we need to have a better definition of the clusters that these primers will be able to define, or to, in the case of bacteria, you don't have all this morphological knowledge, all this taxonomical knowledge. Everything goes, you can use, if your primer says that this is a species, you will say, okay, this is a species because I clustered 97% and we're all good. There's no discussion. So you need to define all these framework before you can start harmonizing the different taxa. And defining this framework for all these taxa together, I think it's very complicated, at least under the current technical and knowledge situation. There's a possibility of changing, for example, the barcodes that we use. Or changing the strategy instead of using amplified barcodes, using a different approach, maybe whole genomes, maybe, I don't know, but you need to change certain things if you want to harmonize taxa. And then you also need a common taxonomic framework for all taxa, which doesn't exist. So reference databases for different groups, and I have been facing that a lot. So the reference database I manage is for Proris. So we have this taxonomic framework that doesn't match the taxonomic framework of those databases that deal with animals, despite all of them are eukaryotes. And that happens for historical reasons. We have been trying to harmonize them in the context of worms and also with GBIF, etc. We are working on this, but it's difficult to fight these inertias that exist within the communities. So yeah, it's a big challenge. And I don't know if we will be able to succeed, but at least we should try to do it, to do the best we can to generate data that's as comparable as possible across taxa.
And we will have fun. Well, we
will have fun. I mean, yeah, we'll have fun.
Emily, you want to add something?
Yeah, or a question maybe to both of you. There's these now tree of life approaches that are maybe a bit emerging, maybe existing for a long time already, that instead of having one universal marker that's going to sequence everything, you're going to have this panel of specific marker for your specific taxonomy groups that then, I guess, ideally, you know specifically what you can and cannot detect and how well. And then combining these, would that be kind of an answer to that? We want to know everything, but we want to know it well and precisely. I think it's something we are going to implement in the next project. And I think it's really interesting to have this ideal combination of precision and scope.
Yeah, there was long ago, we tried to do that for Proris. I mean, Proris, they engulf all the eukaryotic diversity, so they are very diverse. You need many barcodes, and we started to do this. But the problem is that you need to be able to cross-compare these primers. And you need some kind of information that allows you to do that, maybe genomes could be. Now, what we are trying to do is trying to adopt the ribosomal operon as a barcode that includes many barcodes already for both bacteria and eukaryotes, indeed. And also a barcode that has a better phylogenetic resolution that will allow to identify and characterize things that are complicated, such as fungi. And also, it's a barcode that will allow you to map against it many other barcodes to reanalyze the data from the past. With the current technologies, nanopore or PacBio, it's not complicated to generate a whole ribosomal operon in a metabarcoding setting. So for me, that would be the easiest way to move on, rather than combining multiple isolated barcodes that you then cannot cross-compare. That's my opinion.
Michael, do you want to add something?
Yeah, I mean, in the bacterial world, there has been a relatively recent revolution. So historically, bacteria were identified based on the morphology, how they looked, growth media, and now that sequencing genomes has become cheaper and more widespread, this gene-based phylogeny has come in, and that has transformed certainly the bacterial world. I don't know if we're there yet for the eukaryotes or not. Just one comment on, we've mentioned reference databases lots of times. I think there's a really important, there's probably a continuum in how stable those reference databases are. So I'm going to conceptualize a reference database for the British butterflies is probably pretty stable. Once we've got all the barcodes for all the British butterflies, it's pretty solid. The reference database that Javier manages, the PR2 database, that's a database that's going to have to be constantly revised, refreshed, and evaluated. So just from our work that we're literally doing right now, we're interested in groups of parasites that infect diatoms. And so we take metabarcodes, we reference them against the reference database that PR2 that Javier mentioned, and then we have to manually check it. And then what we find is there's already updates since the latest that was done. So I think this, just to be clear, this idea that if we make a reference database, certainly for some groups like protists, that somehow that is fixed and we'll just always be using the same reference. Great, it's always going to have to be updated, but that's a continuum, and maybe butterflies will be fine at some point soon. Yeah.
Well, if I ask the entomologists, they will definitely disagree with you. No, I mean, even in the obvious and visible species, we still discover new species also based on the genomic information. So even there, one species turns out to be 4 species if you're looking at the actual gene code. So I think we are always talking about a flux and always talk about innovation. So we need to be flexible, pragmatic, but flexible. We've chosen for our meta barcoding CO1. I know it's not the most common, especially not in the bacterial world, but at least it's something. Yeah, it will not be great. And of course, we want to know more. Of course, the ecologist screams, now I know the species, but I know nothing about genetic variation, so I need more. And we always need more. Fine. But at some point, how do we get to a point where this is policy, where at least we can say something about our environment? And maybe that's also depending on the question that you ask or the context. Maybe you don't need to recognize all species, but you need to be able to recognize some of them well and track them through time. So as a researcher, I would say, yeah, more is better. Innovation needs to happen all day, every day. But for the world, sometimes different choices, different context.
So in that sense, because I work in the science policy interface, so I'm really interested in landing all of this conversation into, okay, let's talk about how this informs policy, right? So Elaine, what do you think that are the steps to improving like this transference from eDNA data projects, etc., to the policy?
Yeah. I think for any kind of next phase in, challenging biodiversity to become positive, we need to monitor more. And the only way to do that is if we complement the human effort with automation. That's the only way, because we cannot have more ecologists or more specialists, so we need to up our game. That also means using eDNA, not to get the most fine-grained answer, but to at least get an answer given a particular area. So I think it's going to be a mixture of both, and that starts convincing not just the policymakers that this kind of automated way of biodiversity monitoring is highly necessary, but also engaging the communities behind that drive that decision. At least in the Netherlands, I know that the species organizations are fantastic. They have been around forever, but there's also a lot of opposition from them. So let's make sure they understand that we together go into this new phase and we really need each other. So it starts with the humans and it ends with the AI.
So Emily, Javier, Michael, do you have anything else to say about how to transfer eDNA data towards policy?
Yeah, so just my own
Just very
perspective,
quickly.
and I'm happy to be sort of wrong on this, but I often feel like as a scientist, I'm the one directing the conversation at policy, saying, I've got this thing, can I get you to use it? And I would really like it to come in the other direction sometimes. I would really like policy to say to me, these are my needs. Have you got the tools to help me? And certainly in the UK, we don't really have that it's a very one-way street, and you feel like a salesman, you know, trying to sell this thing, and they're going, do I need it? I'm not sure. But really, what would help us if they said to us, we need this, or make this better, or make this cheaper, and we don't have that conversation, certainly in the UK as well as I'm concerned.
Maybe that's a role for Biodiversa.
Javier, do you want to say something, or Emily? Otherwise, I'm going to ask the audience if they have any questions.
Oh yeah, I just wanted to say that I think there are some countries where it is already being recognized as a monitoring tool. I know that the US, I think Japan as well, they have these national strategies for using eDNA. So I think it is in some countries, I think Finland as well. Sweden, yeah. So there are some countries, and I guess it's gonna take up. And I also wanted to mention that there is an, I'm not part of it, but there is an international eDNA standardization task force that is working on these ISO standards for eDNA methods. So I think that will be, once these come out, they're geared towards monitoring agencies, monitoring groups, so that they can get this ISO certification for their, well, they use the ISO certified methods then for their monitoring, and I think that will probably be a big step towards getting into policy.
Thanks, Emily. So we have 3 minutes, I think, in case there's any questions from the audience. Okay, I see one hand at the end. I think that they're going to give you a microphone. If you can also say your name and where you come from.
Yeah, Birgit Gemeinholzer from University Kassel. I'm within the Meta Plant Code project. And thank you for the recommendation concerning standards. And you mentioned the ISO standard, but there are more standards out there. ISO is not open. Are there any recommendations which standards should be implemented?
Recommendations on which, I don't know. I don't know.
There are DIN standards, CERT standard, ISO standards. So, I mean, knowing which would be the best open standard to follow would be a real nice recommendation from Biodiversa Plus.
Yeah. Would be very nice, yeah.
Is there any other question in the audience? Here in the middle.
End of the day, so this is
Rosa, GBIF regional support team. So I really like a comment from Elaine. So maybe I'm just would like to further comment on it. So you suggested what you suggested about selecting the best detected species by DNA could be very nice step for how to communicate with data, with Nature Conservation Administration. But I think we scientists could still continue what you said, this research, improving and so on. And yeah, that's my comment.
No, absolutely. I mean, I work for the Natural History Museum. They want to have it all. But I also sit with people here that are more in the policy and say like, no, Habitat Directive only. I mean, we will meet somewhere and we can do both.
Yeah, just to add, and what we could do to serve is, it's our year. I'm not expert in at all in DNA, but you could select and provide them with a checklist or list with such species and to recommend what to use in their long-term monitoring schemes.
Yeah, absolutely. And I think that's one gap to cross because, well, I'm not entirely sure, but I think the Marine Habitat Directive has only 5 species, of which a bird and a mammal. Where are all the plankton? Where's the, the small stuff that's super important for these ecosystems? The same goes for soil. I mean, sure, I can track a beetle, but there's so many. I think there's 200 different species of ladybugs. 200, right? All the vertebrates together is less than 5% of our biodiversity. So it's insane that we focus so much on birds and plants, sorry. But we need to focus on fungi and microscopic small animals. And we need to focus on invertebrates, insects. That's what we really, I mean, they are also the game changers for their ecosystems. They are the ones responding the quickest. So you also see the positive change quicker if you observe the small things rather than the big animals. So
Maybe sponges too.
Sponges, absolutely, absolutely. Yeah.
Is like, I think that we might have time for just one more question for the audience.
Ruffle, this is your
No,
chance.
there's no more questions. Then I'm going to ask the last question since I have the mic. I just want you to answer, where do you see this in like 20 years? Where do you think that we are going to be in the sense of like, what are the possibilities that you see happening in 20 years?
20 years?
20, 50, 100, I don't know, just the future.
Well, I think Ron actually convinced me AI. Generative AI will make a huge difference also in this field. It will maybe help us select which primers to use or how best to identify species, all the way to doing the dark taxa analysis of all those unknown species we will never see under a microscope. So I think deep learning and AI is going to be a game changer also in this field.
Javier.
Yeah, well, first of all, I think that all the technology that is going to be here in 20 years is going to be different. So no more primers. I cannot envision how it's going to look like, but for sure, these kind of strategies using DNA to monitor biodiversity, they are going to be fully adopted in whatever form they have.
Emily, any insights?
Yeah, I think I'm following that sentiment, is that it's a trusted method. There's still a lot of questioning of why do we detect this, why not this? So my hope would be that there is fully trust, because we know, of course, the limits, and then of course there will be a lot of technological development, but I'd say trust is my hope.
Michael?
I think hopefully there'll be a box that you can just buy.
Yeah.
Just a box. And you just put your sample in, or this box is swimming around in the ocean autonomously, and it's just pinging data back, telling us about what's happening. But I just really hope, so, I mean, obviously we're very Europe-centric right now, but it'd be really great in 20 years' time for this to be really global and for lots of different people utilizing this approach and this technology, and it being standard. So,
Yeah. Well, thank you very much, all of you, for your expertise and deep knowledge on this. Thank you to the audience for not falling asleep after lunch. And
good afternoon, everybody. My name is Giancarlo Rodriguez. I'm the coordinator of the BioJust project. BioJust stands for Biodiversity and Ecosystem Protection driven by environmental justice. And what we are doing in this project is that we are analyzing the environmental justice of several nature-based solutions that expand in different contexts in Europe, from alpine meadows in France to mountain forest in the Canary Islands, and montados in Portugal to páramos in Ecuador and sub-Andean forest in Colombia. And what I'm going to be talking about is two methods that we use in the Colombian case study to actually tackle and include local environmental knowledge and biodiversity in order to understand the environmental justice impacts that the implementation of a protected area had in Colombia. So I'm just going to take you to Colombia. This is the National Regional Park Mississippi Forest. It was created in 2014 by the Regional Environmental Authority, seeking to achieve the goals, the Aichi Target 11, on completing the extension of protected areas in Colombia. The protected area is 2,800 hectares big, and it's declared on private land of campesinos. The reason for declaring this protected area was the excellent state of the forest that were present in these lands, and the importance for biodiversity and water provision to the municipalities that flow along the river that comes from these mountains. The only problem that we had is that the environmental authority that declared this park did not inform any of the campesinos owners that live there. So what we had when we went there, they say, what, protected area? Where, who, what? So we actually had to come up with the first method to visualize how the protected area overlapped with campesino private lands, and to actually try to understand what the cost of the implementation of this protected area meant in terms of land use restrictions, but also the impediment to get loans on plots of land that were inside the protected area. Also another cost was the uncertainty of maybe being evicted from the land that was declared as a protected area. And so what we did is that we start first trying to understand how the protected area was. And we started a mapping exercise where we created this simple map that overlaps the Google terrain photography with the plots of land. You can see the yellow ones. And the polygon of the protected area that was stored in the World Database of the Protected Planet. And we added a feature where you can actually use Avanza maps, and this shows you the location where you're standing. So through this method, it was easy to just start a conversation around how was the procedure of the protected area being achieved, how many meetings were held, who was asked, who was not asked. The good thing about using this feature is that it didn't require to have an internet connection, because there's no reach in these faraway mountains of Colombia. And if we needed to do this with the GPS, then we would have to rent it, but everybody had in their hands a mobile phone. So what we did is that we started walking the plots, and you can see the red line there, trying to understand which agricultural land was inside the park, which agricultural land was outside the park, where the forest was protected. And through this process, we understand that the Missy White Forest was protected due to a combination of several reasons that included, first and outmost, the environmental conscience of the campesinos that understand that water comes from this forest, together with the history of the armed conflict that happened in this region. A lot of the guerrilla from the FARC had a lot of headquarters in this area, and this also meant that there was an agricultural deactivation on the area. Also, the aging population in the rural areas, like in Latin America, also in Europe, contributed to this state of conservation, and the topography, which, believe me, was really hard to get through some of the places, contributed to that. We engaged with youth and women who were actually really good, better than the men, I don't know why, in understanding the maps and how it was like to understand that this three-dimensional mountain in just like a flat screen. And this really proved important for not only understanding the protected area, but to understand how communities in each side of the mountain understand sacred places around the mountain, which they were able to identify. So we did a lot of walks, as you can see here, also with the permission of the army, because we needed not to step on a landmine. And that was really rewarding to actually see the people say, nobody came here to ask me anything about this protected area, and you came here. This is something really, really good. And the second method that we use is that we integrated a method that is called creating visual ethnographies, also through the use of just the video. And we said, okay, just take us to where you see conservation working on your land. People will take us to the creek. We'll explain how calming the water sounds were to their souls, how they connect to the people. Some other people will take us to their bank seats or conservation of land that they have on their plots. And all these visual ethnographies we integrated in a documentary that you can see in this map here, in the QR code here, which is covered, but I can share with you. And it is still in the making. If you want, you can have the link from me and you can see how we integrated, trying to bring up knowledge from the local people that are actually doing the conservation on a day-to-day basis through their practices and local knowledge in a piece that can speak not only to scientists, but to society overall. And this is the story that I wanted to tell you, and thank you very much.
Okay, good morning, everybody. Welcome to day 2 of the science fair. Okay, so today is going to be a sort of more dynamic session. As you can see, there's some tables here, and I'm going to get into what we're going to be doing exactly today. Later, we'll have time for questions, so if you're a bit confused, don't be afraid to ask. So the dynamic of today is this idea of the speedboat. Maybe some of you have heard about this dynamic before, but it's this idea that we have a lighthouse as the goal where we want to reach, or harbor in case so you don't crash into the lighthouse, but you go to the harbor as your goal. Where do you want to be as of now? Then we have anchors that are pulling us down or slowing us to reach our goal. And we have the winds that are pushing us in the direction that we need to be, things that are giving us the push that we need. And we are the boat. So we are the boat and we want to get to the harbor. Gloria, you can help me out if I mess up. So the idea is that we'll have 6 tables. At each table, there is a topic, and these themes come from a survey that we published, a report that we published from a survey last year. Each theme is a challenge that the partners had seen facing in their implementation of novel technologies.
Hello, all. Thank you, especially for all of you who come to the roundtable one. I think that it was a very nice session. So I'm not sure if I will understand the letter of Elena and what we have to write in 30 seconds, but well, there were some main goals that they were repeated over and over in the different sessions regarding table 1. Okay, it was the one regarding the challenge to harmonize the different, to harmonize common methods and protocols for biodiversity monitoring. So one of the recommendations that they were repeated over and over was the importance to agree on an international level on common standards. Okay, it gone from the standard for data collection, but also for data analysis. But there were also some agreements about the importance to make these protocols flexible enough for the different taxa. Something quite, I would say, surprising, it was that in most of the roundtables, most of the people agree that one of the main goals was about the importance to share metadata, to report metadata somehow, and also about the importance to achieve. One of the goals was always to be able to compare the results coming from different monitoring protocols and tools. So some of the wins, some of the positive factors pushing forward these objectives was the growing interest, the growing number of initiatives of international cooperation, including platforms englobing data from different tools, such as GBIF. Also the growing collaboration between researchers and policymakers, but also these initiatives like this call Biodiversa Plus, putting us together, all of us, to discuss some of these challenges. It was one of the main wins, appearing several times. It was also that there are every time more and more protocols and more guidelines being developed for the different taxa, and also the creation of these workshops like today to develop common protocols. And the 5 anchors that appear more often, it was one of the lack of knowledge of existing protocols and the existence of a large number of protocols. So there are for some of the taxa that these protocols maybe they exist, but they are not easy to be found, or they are very specific for certain taxa. So more or less in the same direction, there is a lack of flexibility in some of the methods and guidelines and workflows. So that's not easy to be adapted for different species. But also that there are in some of these processes, there are many stakeholders involved. with different opinion, but also with the, let's say, with the fear to adapt these new protocols about how they can integrate this information with the monitoring program that they have been carried out from 20, 30 years ago. So this is an anchor that is been difficult to harmonize these new monitoring protocols and the existence of too many standards. So I would say that this is more or less in few words for roundtable 1. So thank you, and let's move to the next roundtable 2.
Well, big applause for Christian and Elena for all their help. Great support from Catalan team. Thank you very much on my side and Magina's side as well. Okay, Luis, go on.
Okay, thank you. So I report on table 2 on data management and storage. First of all, I have to thank you all because this has been a fantastic input from all of you. And I have to say I've been surprised by how complementary the different groups have been. So if we hadn't made it in purpose, it wouldn't have been better. So very interesting. I just try to combine a kind of a storyline from the different groups because they have been converging about the main take-home messages. I will be more integrative than Christian here, and different groups have touched different parts of this main idea. So the first one, it's been that in relation to storage and management, we have a problem. We have a complexity problem. We have a lot of data from novel technologies. So one of the main one important ideas have been discussed is that we will have to make sure that to separate workflows and ideas about the use of raw data and the use of processed data from novel technologies. And they will be different workflows that will require different working groups, different descriptions, and different standards. I don't go into the details of the standards, but that's really, in our group, also been an important discussion that has been repeated, the need from standards, that links with Christian has been saying, and it's quite relevant. But in the case of the data management and storage, the standards will have to fit a bit this kind of different workflows that will have different requirements in terms of the managed data and the type of data that it's been needed. So the process data, which will follow procedures more closely related to more traditional methods, but on the other hand, like the management and the storage of raw data that will allow the processing of more complex methods, and also keep the raw information for the advancement in the methods, in the new methods that might be developed in the future, but probably will not be able to do that for all data. So there will have to be a kind of procedure to prioritize and to work together through standards and common processes. We have been discussing a lot about governance and the need to move that together in the different domains. And the last take-home message is that's been very repeated in many groups, especially at the beginning of the sessions. It's been the idea of a common infrastructure, commons initiatives at the European scale especially, but also at the national level, so in terms of responsibility. So create a mandate and a common initiatives that actually allowed or make the development of the standards and the discussions that we have identified before possible. So it's been quite nice to see all this kind of discussion on the different elements that together form a quite nice history. So that's been the take-home message from our side. Thanks.
Thank you very much, Luis and Mireia, for this very helpful help today. Very helpful help. Okay, my English is getting worse. Let's go to Dani and Matthias.
So thank you first to all the people that has contributed to the discussions in table 3. So I don't have this history right now. Only the main ideas that are more shared between people that has contributed in the discussions in our table. So for the lighthouses, one of the main topics that has arisen in every table, I would say, are the standards, the guidelines, the need of harmonization of protocols and metrics for informing this and scaling these novel technologies for policy. Also, the need of demonstration on the reliability of this of the outcomes of these novel technologies. So the products that are aimed to inform this. So we have here an issue on the reliability of these outcomes. Also, a recurrent topic has been the understanding better the policy needs. So we are not there is not a clear opinion on on how to incorporate these needs in research priorities. And at the last but not least, the need of long-term measurements based on shared standard for common use and this long-term reference. About the wins, so most people agree that novel technologies are very appealing for public, scientists, politics. So this is a good opportunity to reinforce the use of these novel technologies. But there is a need of dialogue between these researchers and policymakers, managers, on how to improve the application of these novel technologies. And the need also of success stories and case studies, examples to show the real impact and the real application of these novel technologies. These are the wins and the anchors. So the immaturity of novel technologies and the limited capacities from manufacturers to provide and to support the upscaling of the application in the real world. Also, the lack of flexibility. So there is this closed designs and lack of flexibility. And also another anchor is the loss of background knowledge and expertise to adequately interpret the data produced using these novel technologies. And finally, the lack of long-term support, funding, etc., and the need of short-term urgent results coming from policy. So policy is asking for, we need this right now, and novel technologies so are immature and needs more time to produce this relevant information. And this is all. So thank you very much, all. And
thank you very much, Danny and Matthias, for your help today. Let's go with Gerard. And who else was here?
Thank you very much. Thank you.
And Guillaume, of course.
Yeah, we'd like to thank you, everybody who was in table one, and also to the support of Guillaume that has been invaluable. Over the groups has been some ideas that are overlapping. The topics somehow they move to one side to the other side. I will try to, our group, my group was focused on the cost and expertise required to be implemented. And one of the things that was arise more commonly that is we, as a lighthouse, that we need more people with new expertise in these topics, both in taxonomic, but also in all the technological issues that they are coupled with all these new novel technologies. So we identify we have a lack of this expertise. Some of the expertise Turkey, they move to other sectors that they have higher salaries. So it's difficult to attract techie people to our side. But we were thinking as a wins that we can promote, and there is some initiative that specific training careers in this topic to create new jobs opportunities linking the new technologies to biodiversity monitoring. One of the strengths of novel technologies is at the end, some of these, it can increase cost efficiency of the monitoring. So this can be very linked to the policy needs, because sometimes policy needs quick response on some of the questions. So we saw as a cost efficient in terms of economic and response that a win is that novel technologies can shorten this from data collection to reporting. They shorten the time that normally politicians need. Another thing that we have and it's important, it's that we need common hardware and software protocols in terms not only in the data that with the common specifications to guarantee the interoperability and the results of the products. So this can reduce the cost and also the needs of expertise. And there is other fields of industry. They have common, the ISOs where all the specifications on how, I don't know, microphone sensors and all these things. So we have identified there's a lack on that. So this fragmentation is a pitfall. And if we increase and we give not only protocols of the methodology, but also in all these parts will reduce cost. And the last but not least important, there's a differences between countries in terms of funding and speed on the implementations, but thanks we are in Europe and probably is one of parts of the world that we have a willingness to collaborate between countries. So this is a nice opportunity to share infrastructures, national coordination centers. So having common protocols and the standards at the end will reduce cost and make easier all our jobs. Thank you.
Thank you again, Gerard and Guillaume, from the help today. We will continue with Maria. 2 more tables to go. Just quick words to have the closure, and then we will go to lunch. Just a couple of minutes extra, and we will be there.
Nice. Yeah, thank you very much. And thank you very much to everyone for your contributions. We were the topic of how to integrate all the historical and novel technology data and methods. Of course, it was really case specific when we think about which type of historical data and which type of novel technologies are we talking about and how to integrate them. But to summarize, for example, one of the common, more repeated goals that we are moving towards is to really have long-term observations to really identify the impacts of global changes, for example. And then at the same time, this brings us to have this availability of data that it's really open for everyone, for policy, for research, and then to use also for the private sector. But in turn, like these have like a certain anchors attached to, no? That it's this lack of data standardization, and then is also maybe the lack of accessibility to this data and how findable it is. Like how do we know where is this historical data and how we can make use of it? And at the same time, how is this data shared? And it was a common discussion as well, how reluctant many sectors and groups are towards their data, no? Of making it available. We also discuss a lot about costs, for example, when it comes to all this digitalization of like historical data. And then also identified this, the taxonomical changes that has happened, like comparing historical data with currently data. And that also brings us to the lack of expertise of taxonomy, which in turn, it can turn into a main goal itself, no? That it's this bigger goal of like way to keep track of changes in taxonomy. But we also identified those wins related to, so for example, as we have been discussed in the other topics, the need for metadata standards, but also how AI was one of the topics that was more commonly like mentioned, that it can help to digitalize and reduce those costs that we were associating to. There is a need as well for the historical and data validation, and then to check how good those different data sets are, and then the possibility to integrate them or not. And then finally, like to have the open data repositories, and it was mentioned several times again, like the GBIF availability, like Darwin Core and its extensions, that it can also add the reports, and then how we can make that information available so everyone knows about it as well. Thank you very much to everyone.
Thank you very much, Maria and Ana Costa, for your amazing contribution. Now we will welcome Ana de las Heras, which has been with Iris there.
Okay, hi, hello everyone. I'm gonna try to make this as quick as possible. We're all hungry, so well, thank you so much for everyone who participated in the table, and thank you so much for Iris for taking notes, which was amazing to have her. So I'm gonna do this in shopping list style. So very brief. So basically, our lighthouse of main goals were a good science policy interface that should be inclusive, standardized data, a unified message, and then a multi-directional approach to be able to include all different actors and to have good knowledge transfer maybe positions that can be adaptable to the different needs. And then there was a big one, which was the mainstreaming of biodiversity. And then our anchors, they were on language, both from a technical language perspective and then from a geographical part. So in Europe, for example, we have a lot of different languages. Then we have the compatibility of data with all the different methods, especially when we travel across time. How do we move from one system to the next system? And then there was timing, which is both timing in terms of needs. So different types of actors need different type of information at different times. So how do we deal with that? No? And then finally, there was communication and coordination amongst all of these actors. But then thankfully, we have all of the wins or all of these things that are going to help us achieve this goal despite of the anchors. And for that, we had tools like policy briefs that can be very quick to access this information. Then we had good capacity buildings with seminars and workshops. These were the two most mentioned methods to be able to do that. And then finally, we have good communication of these needs. So really emphasis on how we can transmit these ideas and to incorporate all of this social science or all of these different disciplines into it, either with translators or with transfer officers. And my favorite idea was doing a talk show about biodiversity. So, but this was a bit of what we discussed this morning. And of course, around the topics of knowledge transfer and capacity building, which are very different, but at the end of the day, they are quite interconnected. So thank you very much.
We welcome Guillaume to talk a little bit about future steps.
Hello, everyone. It's very nice to have such a community of practice here about monitoring. So I'm very pleased in the name of Biodiversa Plus to invite you all next year in Montpellier, which is not too far from here, just 2 train stop station, to what we call the first European Conference on Biodiversity Monitoring. It will be much more than a conference. It will be a full week of discussion and sharing practice. We will have guest speaker, parallel session talk as usual, but also hands-on and workshop to put in practice our idea and to share the way we work. And we will also take care to invite decision maker to make the bridge between science and decision makers. So we expect to have it early on May 2026. We expect to have 400 people coming and sharing ideas. That's a brand new conference. And in the idea, we expect it to run forever from now and organized at the beginning by Biodiversa Plus, but in fine by whoever wants to organize such a conference for the community and by the community. So we'll take care of the organization Biodiversa Plus, but we will also invite you, the head of communities, to participate also in the organization of such an event. So thanks for these few days that are a small part of what we will have in Montpellier next year.
Exciting for next year. Now we welcome, thank you, Guillaume, for everything, and welcome Ron on stage to say a few words for the closure of the session.
Just a few words from my side, from Biodiversa, and while I'm talking, I would like to invite all the organizers here on stage so we can properly thank you. So please. So we can acknowledge everyone who has had input in this organization. For Biodiversa, it's very clear as an organization that the strength of Biodiversa as an organization really is reflected in the breadth and the strength of the partners that are involved in it. And that online clearly is not a proper substitute for involvement, as you can see in a science fair like this. This is the first time we actually had a science fair. I found it really enjoyable. There's lots of interaction and discussion, and I really appreciated the presence of companies. Actually, the timing was perfect also because we'll have a workshop with companies later, so they could be involved in the discussions also today, giving their views and adding their views to the mix. So that was really perfect. It's been a ton of work together with the OT from Biodiversa, but I can see the stress already falling from their faces now that it's over. And no, thank you very much. I mean, it's really, really well organized in a wonderful, it's a super beautiful location until the rain today. We had a lot of luck with the weather now. It was wonderful. And the dinner also late yesterday was a super fun place to have it. So very much appreciated. Maybe next time again, but no, but we appreciate this very much. So just to round off, yeah, we will evaluate all of this because we would like to have your input of what you really liked best, or what things you would miss in something like this. Because as Guillaume just said, we will have a biodiversity monitoring week in 2026, and we also have one planned for 2028. Beyond that, we can't look as Biodiversa because formally the current Biodiversa ends in 2028. But we hope to establish that as something that every 2 years you have a biodiversity monitoring week with a meeting and workshops, as was just outlined by Guillaume. This whole meeting did give me one overall thought, because it's on new technologies and how it influences us and what it does with our work. Funny enough, at my organization, we had a similar discussion, and this was on biotechnology, and was asking, what are the new technologies and what are the major developments and how does that influence us? And AI also came along. And then when you start to list what new technologies can do, collect tons of data, and then AI will interpret everything. AI can develop the protocols for you. AI can do this, that, and the other. One question that we had, and we're going to discuss next week with a lot of scientists. So what does the future ecologist look like? What will the world look like for the young ecologists that are studying today? What will their actual work be? Because we have this vision of people going in the field with a butterfly net and collecting stuff and looking at things. But as we heard in the last couple of days, so much is going to be automated. There will be machines everywhere. There will be AI to interpret it. So that's just a thought I would like to leave you with. What will an ecologist be like in the next 10 years? And well, maybe we'll meet each other at a biodiversity monitoring week in 10 years and discuss what happened with us. So thank you very much for now.